Good morning, everybody. Welcome to ROV Sebastian, dive 955, live from research vessel Falcor 2. We've just put the vehicle in the water, got the last of the floats off, last of the floats on, sorry. And we are heading down to a depth of approximately 1,160 meters today. So we're going to turn the mics off now, let you enjoy the descent in peace and quiet. And we'll turn the mics back on. We'll have a scientist to talk to you once we get to the bottom. Okay, enjoy the dive.
No? Okay. Hi, everyone. My name is Kyle Foster, and I'm a student at the University of the West Indies, St. Augustine campus, studying deep sea biology. And today we are conducting an 11-hour dive. We'll be exploring a mound, which we suspect might be a mud volcano. And we'll be keeping an eye out for bubbles or methane that's being emitted from the seafloor. So this area has been mapped. During this expedition, we've been conducting mapping using a multi-beam echo sounder. So that instrument essentially emits sound towards the seafloor, and it bounces back and paints a picture of what that seafloor looks like. So the same way how we have mountains and valleys on land, we also have similar features underwater. And associated with these different features are different types of biodiversity. So while mapping, we noticed, you know, these areas that have been unexplored virtually, visually. So we're here to see if we see anything interesting and confirm flows of bubbles as well. So the first thing we're going to do once we touch down on the seafloor is conduct some heat flow measurements.
Is this area good here for landing?
Yeah, here's good. Yeah. And in the room with us, we have Rob Perrin and Rachel Lauer, both geophysicists, so they will be keeping an eye on the instruments and essentially, taking measurements here. So a mud volcano might be more exciting than what you might be imagining. I mean, of course, they are quite interesting features, but there wouldn't essentially be, mass eruptions of anything in particular. You will see hopefully some bubbling coming up, and of course, with this type of feature, we have chemosynthetic organisms associated with them. So that would be organisms that use the chemicals that are being emitted and transforming that into energy. So the same way that we have plants on land transforming sunlight into energy, we have animals in the deep sea away from the sunlight in the deep dark conducting these similar processes using chemicals. So while it might not be like as exciting, we will hopefully see some interesting biodiversity around, which is exciting in itself.
What's going on with the sit cam? What do you mean?
It was just the iris was so open.
So what we're taking out here is known as Excalibur. So it's a device that we push down into the seafloor to basically test, to test how deep the seafloor is. We want about 60 centimeters without hitting any hard substrates. Oh, it came off.
The fireboats? What is that, the rock box?
We take Excalibur and we push it as far as we can down into the sediment. And you can see the sediment around here is quite soft. Maybe go a bit further. Good?
Okay.
Yeah.
I'm recording. You can push the probe in. Yeah.
Okay.
So this one is
Probe down.
HF 26. No, just the name. What's the dive number?
This is 955.
Weird.
Yeah, hopefully it sorts itself out.
I don't ever remember seeing that problem.
This is very similar to what happened on the last one. Only it was just 3 that time.
Now it's 4. 5. You know, you'd think with the higher precision that you'd see less of this, right?
Yeah, I don't know. Well, like 1 and 9 are funny because I'll calibrate them and then they'll just kind of like jump out of calibration. And then I'll
Yeah.
calibrate again and bring it together and then they'll kind of slowly drift out.
Can you?
So among
can
heat
we-
flow measurements, we'll
would it
also
possible
be
to give us some ideas
conducting
for-
biological sampling of
Yeah.
any
Is there gonna be some
interesting
for
and
us in
unique
the
species that we come
Mhm.
across,
That's so
which is
weird.
particularly important
Like,
because
four of them
we
behaving like you would expect,
are
and the
also
others are, like,
conducting
together.
genetic studies. We're also taking these for taxonomic vouchers, and all of the samples that are being collected would be housed at the UAE Zoological Museum. Previously, any deep sea expeditions that would have taken place, unfortunately, very few of the samples would have been left in Trinidad and Tobago, which is important for locals to be able to connect with their deep sea biodiversity, but also being able to understand these organisms on a more genetic level. So it's important that these things are kept locally. So collecting samples, keeping an eye out for methane seeps and bubbling. Heat flow measurements are just a few things that we'll be doing today. And this is a site that has not been visually explored before. So, yeah. I always find it so interesting when we zoom in on like the sediment. You see so much more. Like, what are these? these dots that we can just see here in the sediment.
What is that?
Those there. I don't know.
What is it?
Oh, I don't know. I'm also thinking the same thing. Little shrimp. Oh yes, another thing is that we also can collect sediment cores. So there's also a lot that's going on beneath the surface that we might not be able to see. So we conduct some coring, and we have a team that sifts through the sediment for macrofauna. And we're also seeing, lots of bits of sargassum seaweed as well. If you're familiar with the Caribbean, or if you live in the Caribbean and Europe at this time, many of you would know how the sargassum seaweed has essentially inundated many of the beaches across the region since like 2011. And I'm always interested to see how deep sea species in particular interact with sargassum seaweed. We've come across quite a few large mats down here. The majority of them don't really have any species interacting with them. But there was one where we saw like a whole host of amphipods just living in and around it, and as a result, some crustaceans and other species, larger species that might have possibly been feeding on the amphipods. That's a little, is that a squat lobster? A very tiny squat lobster. And yeah, like when you zoom out, it just looks like it blends in with the rest of the white dots. So there's just so much diversity that if you don't take the time to, pause and investigate a bit, you just kind of pass over.
It's like low.
Yeah, we're seeing a lot of little tiny shrimp. Yeah, this one. I mean, this looked pretty interesting, to be fair. Even like right below the shrimp is well, like a tiny, maybe like polychaete, some type of worm. It's just like burrowing into the sediment.
Kingdom's on fire today.
Oh yeah.
See anything on the sun?
It's fun, Jerry.
Beautiful here.
That's wonderful.
Nothing.
Yeah. Yeah, man.
Well, we're not on the mud volcano yet. We'll get, I promise. Have a good night, morning. Yeah.
Good morning, everyone. My name is Jamie. I am taking over from my fellow colleague Kyle. We are both students together at the University of the West Indies. We're just finishing our heat flow measurement. Should be done in a couple minutes.
Okay, we're done.
Oh, we're done now.
Yeah.
Perfect. Thank you.
26. Sorry.
Oh,
Okay,
like
track,
Do
man.
26 and 26.
yeah. Morning, Sunny.
Morning, how are you?
Good. Yeah, and as we come up, we are going to do a video transect for 300 meters
Okay.
on the way to waypoint 2.
Headless chicken monster.
I think so, yeah. Yeah, it looks like one. Yeah, very translucent.
Cool. Is that a headless chicken monster?
Yeah, it looks so.
Yeah.
Headless chicken monster. Yeah.
Okay. So you take this caliper up. Yeah, because it's different. Last minute changes. Following, right? Is that at 300 meters?
Yeah.
I don't know what the change. Oh.
Oh, thank you.
Give me the other one. It's
Oh.
wrong.
Thank you. Yeah, so 300 meters.
This.
Same like flashing lights beginning
Oh.
area.
Okay.
Yeah.
Yes.
So we did a heat flow measurement as soon as we touched down. And now we're gonna make our way to a mound that we want to explore that potentially may, we saw bubbles while mapping, so it may be a seep. We are not sure. But on our way to the mound, we're gonna conduct a video transect. And then when we reach the mound at waypoint 2, we will then take a second heat flow.
Measure 300 meters from this.
And then from there, we will then begin to climb the mound and explore.
bubbles with the level.
What?
You go?
The bubbles.
No, I mean on the.
You let me know when it's
I
not
don't
done.
know. It doesn't do very well on the.
That's not-
obviously
Yeah,
not
yeah,
really
I think
designed
it-
to have two sets of
Get
waypoints
everything
on.
ready.
So you're far better using
Doing
like
it.
using
Okay.
Thank you.
frame
Charlotte, start and transect.
science.
Go ahead.
Yeah.
You get a lot of sort of waypoints.
The red or the bubble.
Away we go.
On Fideras.
Yeah.
Yeah, I know, but
There's science.
Yeah.
There is fine.
Yeah.
Mhm.
That's right.
So, do we have the 100-point marks? So,
Yeah. Well, maybe not. For flashing the lights. We're gonna-
100.
Bridge orbit.
Orbit bridge?
Can start moving the vessel.
The shadows.
If we're able to get rid of the shadows.
Seems to be yeah.
Is that better? Oh, you see my baby. See big ones now.
Your first? Order.
Oh boy. Core. Core.
Cool, huh?
K-A. Like Noah, but I guess with a K. It's a Hawaiian name.
Sounds like it.
Yeah.
Your husband or
He was so close. Very
you know Keith Lewis?
Yeah. There you go. Okay,
Me
keep
and
this
he
cool.
was trees.
Ah. Yeah, makes sense.
So, you know, Sebastian and Christian and all that fellas.
Mm. Yeah, small community.
Yeah.
So we're currently at a depth of just under 1,160. Trinidad's waters go down to about 4,000 meters, which we did go on and explore early in the expedition. But moving forward, we're actually going to be exploring slightly shallower waters. So this is the deepest that we'll be for the remaining of the expedition.
Okay, we. Yeah, I remember when Mike did those last night.
Yeah, flies by.
Yeah.
We've done some deeper measurements. So sorry.
Oh, how are you?
9 and 10. It'll be 10 and 11. In September.
Back to back.
Yeah.
Pressure.
Cheerio.
And they're home in Trinidad?
Ethan and Scarlett.
I like the name Scarlet. I'm impressed. I think SDA coming up soon.
Yeah, on Sunray coming too. After the next measurement. Sure. Oh, sure. Okay. Like age, probably. Sure. Okay, 100.
It's doing heat flow at waypoint 2. Do you have any heat flow back for it?
Just the one. And it's only half the one.
Is it current strong versus normal?
That's okay, right? Why you ask?
I was curious. Everything's just floating in it.
It slowed me down a little bit. I was on point through that. Down.
There are a lot of jellies in the water.
Or the bridge, you don't undertake waypoint, are you happy with stuff?
Copy that.
Put it to waypoint 2. All the way up to waypoint 2.
So
New waypoint.
before this expedition aboard Schmidt, Falco 2, we have actually only studied about 0.001% of Trinidad and Tobago's deep sea. So I'm really excited to see what that number becomes after this expedition. And we also have a record of 451 species previously, and I'm also curious to see how much that jumps from this expedition alone. From our statistical analysis, on being on the conservative side, we believe that we're missing about 70% of species from our records, but it could be even more. We've actually been seeing a lot of these large sea cucumbers at different sites. Good. Ah.
Okay, I was on, but
100 left.
That's the one. Right, so that means we sort of know like what conditions. So we should yeah. Yeah.
Strong warrior.
Okay, yeah, alright.
But it's also a type of tree in Hawaii.
Okay.
His dad was set on that. That's my hope. If we have a girl.
Unless he has a Hawaiian.
No, he got the first one. I got the second one. So the mound we're going to could be a mud volcano. We don't know.
And been on the street times.
And we actually were on the mud
I
volcano.
didn't see it.
It actually just looks like a mound. It really looks, I don't think it looks to the front. But I think it was a mud volcano.
And when people said volcano, they're expecting
Like eruptions
caldera,
and like
sea
yeah.
things popping out.
Same. It's a little underwhelming.
Yeah.
I know. What I know scales.
So what gonna be a takeaways from being on this lesson? Something you see it could be
Yeah.
you
I-
can do regular.
Regular-ish. Well, I have a little kid. I think the biggest takeaway is how much collaboration it takes to achieve goals. Like from the science and the team. It's like it takes a lot of people to make it happen.
Yeah.
Like one person can't do it at all. But yeah, it's a lot of teamwork.
So what was your line of studies at?
So I'm a master's student at uni. It's an MPhil. I'm studying, I guess it's environmental / deep sea biology under DIVA. So yeah, this is my first expedition. Research.
Couple of years in really, I mean, science.
Maybe in my fifth day. If ever. A lot of learning to get there. outreach.
Yeah.
Yeah, I think so. I mean, all of the news stations have to cover it. If Daddy's laptop's with you. That's for the first half of the expedition. You guys are gonna get to drive it. Or
Yeah.
and then you guys do it. It's switched.
You living up to expectations.
Good show.
Just zoom up.
It's probably a little tripod fish.
Okay. 100.
That's the end?
Oh, that was
300.
300.
Yeah.
Are we still out?
Charlotte. Yeah, it's the end of transect.
We still have a release the
Waypoint
two,
two.
right?
Yeah, yeah, but it won't be transect.
Right.
Hit the ground.
Yeah. 300 of the.
Yeah.
So that's 300, right?
Yes.
Yeah. So if we see something, we look at it now.
Yeah, yeah, we can. And then when we reach waypoint 2, we're going to do another heat flow.
Okay.
And we don't have any like locations that somebody has gone to. Especially if it's like 2 a.m. If it's in the morning, I think. It's just at 2 a.m. I can't say anything about that. Because it's gonna be.
How come we like to start like on the soft sediment bottom and then make our way up the mound instead of just starting on the mound?
For the heat flow.
Just for heat flow. I was wondering, there's always also like UK biology.
Oh, yeah. Well, I mean, I think for them, going down is not ideal.
So we have to
So
go up.
we'd like to go up instead.
And yeah, then you need soft sediment for heat flow.
Yeah. And then we start a little ways out, so we want to see some change as we approach
The mound.
yeah.
Yeah, that's fine. See that. If we see another one, I'll stop and zoom in.
No, we're not stopping.
Okay.
We're going to wait back there.
All right.
No stopping. People make like that.
Yeah.
I mean, no stopping.
Yeah, we're almost there, I think.
Yeah.
So right now we're on the soft sedimented floor before we start climbing the mound. And the community seems to be made up, from what we've seen, of urchins, some shrimp. We've seen a couple of tripod fish, some halo sawfish, little bits of sargassum dotted here and there. And we are currently exploring Trinidad and Tobago. We're east of Tobago right now, a bit further out. And yesterday on our dive, we saw some amazing corals, a beautiful coral garden, a small seep at the end. We saw a shark, or a false cat shark. Probably one of the coolest ones. Interesting.
We can take the measurements.
You want to take it here?
Well, I'm just starting to
See some shells, yeah, and rocks.
Yeah.
You able to pause?
Yeah.
Yeah, 'cause it's about to get rocky up front. Yeah. So yeah, we can take it here once it's soft.
Bridge ROV, can you stop the vessel, please?
Over to the stop.
So we're just gonna try and take our heat flow measurement here while the sediment is still soft, because it's looking like it might potentially get a bit bumpy up ahead. Is that fine?
Okay, I'm recording. You can insert it when ready.
Okay, and save it now. Okay, full titration.
Okay.
Yeah, it's all the way.
It is.
Yeah.
Okay. Four to nine, but they're behaving properly this time.
1, 2, 3, none.
Yeah, I know, but yeah, but 4 through 9 are there. So.
But I mean, it's not all the way down in the track.
Oh, the tracks.
Yeah.
I see.
Actually, is lined up at the bottom of the track, so maybe we can check. No, the heat flow program.
That's all it's going down there. No, no.
Did it
Keep
get
it.
did it get moved on the?
No, I don't think so. Unless when they put it back on, they didn't put it.
Yeah, but we've used it a couple of times. It's just it looked like it usually lines up. Anyway, we can check it when it comes back. It's just not hitting the first 3 thermistors.
To go down more.
Yeah, it
What?
used to. That's why I'm asking because I've been watching this for 3 weeks now, and
Yeah.
it goes down.
Okay, well, I
further.
think what happened when we took it off,
Yeah.
they didn't put it back. It should have been on the lower hole there.
Okay.
So
we
it'll
can-
been.
Okay.
So I think when we come on deck, we'll have to rectify that.
Okay. Cool.
The gradient seems higher. The gradient seems higher.
Is it? Cool.
So you can watch them in your
Has light?
Yeah. Yeah, the baby monitor you can also watch on the app.
Like it.
You can move it around.
What's that signal
Crazy.
jump?
What that was. Did you wait one?
So who's up in here and has them take care?
My parents.
Your parents?
We live next door to them,
No.
like attached to their house. And then his mom helps as well.
Yeah, well,
There's a lot of people
Is
taking
this-
care of him. And then my cousin, who's a school teacher, also helps out while she's on vacation. It's nice.
What I saw before coming in here.
About it? Yeah. Yeah, I've like watched some of the expeditions and known some people who've come in. And then Diva's been talking about it for 2 years, so when applying.
So a video came up on the feed yesterday.
You know what's this one?
No, about the normal stuff is here.
Even all that
Yeah.
in 2014, yeah.
Yeah.
They came for 3 days on their way to Grenada.
Judy.
Good.
Okay.
Yeah.
Where are you looking at?
Right, 6 o'clock from your arm. For example. Use that
Shell upside down.
Yeah.
Very
I mean, I'm also seeing some bacterial mass pop
I
around.
know, but like that.
Go up.
These are rocks.
Little rocks.
Ones to the left. Yeah, see this one? Okay. Right on the edge of the street.
Yeah, that one's a
No,
rock.
no, up.
Oh.
See it?
I'm
Right.
guessing that one.
Right where your laser's now. Way to be fossilized. Look at the technology.
Still very tiny. Actual seafloor probe.
Now you can sleep. Right, so now that we've taken both our heat flow measurements, we can start exploring the mound.
Certainly a lot less seaweed on this side. Remember the
Yeah.
abyssal ones on the other side is a ton.
That's what I was actually thinking, that
Yeah.
it's interesting the difference in patchiness.
Yeah. Yeah.
It's about to get a little rocky. what lobsters, corals.
Hey, not local. AN. 60, 65. I want to keep
All
that.
shells.
Yeah,
All
so it's a mixture of shells and rocks, or just shells?
both, I think.
Both.
So these are really old shells. They're getting oxidized.
It looks like it got steep.
Yeah.
Are we able to look ahead a little more? Just not 3D.
Yo.
I mean, I can see it
Look
there.
at this one.
Yeah. Yeah, that one's more like, I guess,
The instructions
precision.
with that one is just above the
Ah, okay.
front
Okay.
port.
Alright. That's the steep mound.
Definitely got rocky right before they sampled. Sticky sample.
Yeah.
Yeah.
Waypoint two? Yeah.
So you want me explore on or start moving to 3?
I mean, we can explore while we make our way to 3, I believe. Yeah, like head there.
Because there's something
In front
yeah,
of
in front
head.
of us there.
Yeah, let's see what's ahead first. It's the rocks. It's,
Why is it not called a starfish anymore?
I think it is. I think it's both.
Uh, no, we're gonna be going up promenade.
Yeah.
I'm just seeing what's out on the
On the sonar before we head up.
Yeah.
We never got a piece of that coil yesterday, the white one. We see it.
The white coral.
Like this one.
I'm not sure which one it is. Oh, the big one, you mean?
Yeah.
Yeah.
If it's the same species, I think it is.
No, just
Yeah.
remain there for. I think we're gonna start going up. No.
Yeah, we'll start gonna. I just want to see what.
This way, right?
No, we're.
Do you think it's
we'll be
this
going-
coral?
Yeah, but when you go up, should we go
Okay.
right?
It'll be on the left.
Look how they're broken
It'll
pieces
stay on
of
the
it.
left of the line.
Interesting.
It's not better just for you for
That's good.
this position
Hmm?
instead of.
Yeah.
No,
We keep going.
keep going.
Yeah. Just a little bit, and then we can head to waypoint 3. Those old tube worms. And bacterial mats. Yeah, something massive. It's like a massive wall. Yeah, rock slab.
Oh, look at all the corals there.
Wow.
So let's take a
We're gonna take a look at that. Yeah. When we get up. Right in time for Eugenia. Coral lover.
Look at all the worms done.
Yeah.
Oh, this is different.
Yeah, these are different. Wow. Good idea to keep going there.
Go ahead, Bridge.
Yeah, for now.
Yes,
Yeah, we're
happy
close.
for now.
So if we're able to get like good zooms. Wow, hovering.
See how we have all these two worms right next to the coral?
Well,
Jamie?
Yeah. There? Okay.
Zoom into that.
You want to zoom into like
See?
that patch at the left one. Yeah.
It's like a mixed.
Whoa. Look at those guys. Yeah, they're tube worms. But I don't know what the white, and I don't know what the white stuff is. It looks spongy.
So maybe we can hold it there for a bit.
Yeah, we'll hold it. We'll hold it. You want to zoom in and hold it for a bit?
Oh, it's fine.
Yeah. Yeah, and then we just want to hold it for a bit.
Just give me about 5 meters out. Yeah, 5 meters.
Leaving when it gets fun. Even behind them.
Zero.
Yeah.
So we just explored if we see something like that, so both of the waypoint.
Good.
Hi guys, morning or good night, wherever you are. My name is Eugenia Thomas and I will be narrating for you for the next hour. I am a marine biologist and I'm also a MPhil student at UE St. Augustine studying deep sea waterfalls and deep sea debris. And as you have heard this today we are exploring site 6, and basically on this dive we are bubble hunting. We are looking for seeps. So we have just passed waypoint 2, and we're basically following any signs of seepage. That means broken shells, tube worms, etc. So we're just under the mound, and we're seeing a lot of corals here. We're seeing some tube worms that are intertwined with the corals as well. So that is a good sign that there is seepage nearby. So many animals that attach themselves to corals. Seeing some broken shells. Yeah, we can.
Okay.
Yeah.
Right
Oh.
on the side of the check that out.
Yeah, so this part is seeing all the shells there. Alright, so we're seeing some bacterial mats, we're seeing some more chemosynthetic shells. And bacterial mat is a good sign of methane seeps nearby, because at methane seeps you have a lot of hydrogen carbons that are being released. And with this you have high microbial life.
Also
Ooh,
down here, right?
that
So,
is-
these stable.
That is nice. Dense population of tube worms here. It's almost like though they form their own ecosystem. I see some bacterial mats. Again, this is formed when you have so many microbes that are in the water column as a result of the hydrogen sulfide being produced. And these feather-like structures you see in here are actually living tube worms, and they basically trap any organic matter or methane that is in the water column, and that's how they get their energy. And this is a prime example of how chemosynthetic work in the deep sea. So we're just going to stay here, look at the tube worms in action. Right. Yeah, and then we have a living mussel at the end, at your far bottom area. Yeah. Waypoints, yeah.
No, just right click. Add a marker. And then label it.
So they're just watching these organisms up close.
Tube worms and corals. Worms, W-O-R.
Yeah.
O-R.
And so if you guys are wondering, how you can tell the difference between the living and dead shells. Well,
WOR.
in these areas, we typically see two types of shells. We see the Patermilias and the Gigantus. And so those are usually brownish, yellow, blackish in color. And so when they're no longer alive, they lose that color and they look pale, they look white. For example, you have the living one that is brown, and then you have the dead one right below it that's pale in color. You also seeing some coral branches on the rock as well, squat lobster. And this squat lobster its color is a bit different as to the ones we normally see. This one's kind of purplish. Yeah, definitely a different kind to what we usually see. Also seeing some sponges as well, some benthic ctenophore. Oops. Seeing some other tube worms. And so this one is very much similar to the ones we just saw. See some sponges. So basically, when they're not filter feeding, they usually look like this, you know. And this is another example of how a dead shell looks. It starts to lose its color, and it's no longer a whole organism. You don't have, you know, basically a complete shell. So
White stuff.
those are bacteria mat and some sponges as well.
Anything else you want to look at there?
No,
Right,
that's
zoom in.
pretty much talk about everything. Yeah. That's it. Yeah. What is that? I think this is a sponge as well. No, the yellow one. Yeah. So this is our other type of sponge we're seeing. So this is our other sponge we're seeing as well. And you're also seeing some bacterial mat forming around the old tube bones. So and this is just a close-up of just the intricate details that surround signs of methane seeps.
Happy?
Yeah. You could go ahead and run on.
What was that?
That's great. Thanks.
So we're on the move again. Hoping to find large mussel beds, bubbles, but we're close given the evidence of tube bones and bacterial mats and shells that we are seeing. And our scientists would have spent hours mapping these areas. So it's always good to see So far on our dive, we've seen some octocorals, and we've seen the common species, we've seen some shrimps, some fish. Also seen some small jellies as well. And unlike other dives, we haven't seen much sargassum.
Yeah. Oh, just trying to get something on the sonar there. We're okay right now.
Seen some more corals, some broken ones that are on the floor. Also seen some more of the old tube worms, sea anemone, squat lobsters. So
Okay.
Yeah.
That's probably McDonald's week.
yeah.
Pre-gyrovic.
Oh, David?
You can start moving the vessel to the right.
A lot of sediment being kicked up here, and it could be as a result of underwater currents, or it could be an animal that was recently passed by, but I think it's more on the currents.
But it was interesting.
You see more rocks here, and I believe some of these rocks are mostly carbonate rocks. I mean, I'm not a geologist, but these type of rocks are associated in areas that are nearby to seeps.
Yeah, that's the one I did, yeah.
Yeah, you see, I did the inside. Yeah, thanks. It's good to go though, yeah?
Oh yeah, it's dry and everything, sorry. Yeah,
I've seen a sea cucumber.
The front,
This is what would be stripped off.
good to go as in it's dry.
Yeah. You just want to listen for a quick second.
Turn the laser on there, Marcella.
So we don't usually see this type of sea cucumber on our dives. I mean, we have seen it, but not this one usually. It's pretty cool. It's amazing how you see in all the internal organs. And sea cucumbers are a great bioturbators, meaning that they feed on the organic matter and any decomposition in the sediment. You also see in our cup corals as well. And so they basically feed on the sand and then spit it back out, essentially. And so you have clean sand being returned back into the environment. And so these organisms are good for nutrient cycling in the deep sea. So coming up on another huge rock, and of course we can expect to see old tube booms that are encrusted on the rock, and of course corals. Sorry? Or go more to the left?
No, just look down there, listen.
Oh. Yes, so we're seeing some bacterial matter again. So closer to the left hand side.
Yeah. I saw this fish here.
Yeah.
Looking peculiar.
This one here?
Yeah.
Mm-hmm.
Alright, deal.
It's alright, dude. I'm really a fish ID expert.
That's a rat tail, right?
So many interesting fish we see in the deep sea. So we're seeing some bacterial mats again, and we're seeing some shells, seeing some tube worms. So we're nearby, but we just need to follow the evidence. Seeing some more broken shells here. Seeing some old tube worms that are intertwined with some corals. Seeing a huge coral. And what is that? Octopus? No? Oh, well, some fish.
What do you call it?
What?
What? Trinocops, I think.
And trinocops.
Yeah.
Oh, this is so cute. This fish is
This is one of
adorable.
Juliana's favorite fish.
Oh,
What, tuna cops?
Yeah.
look at that. So it's a
Look at
tuna.
that,
Is it tuna cups?
Trinocop. What
True.
do you call it?
That's her favourite type.
Yeah.
Okay,
Similar,
so
Say it again, Jimbo.
true.
There you go.
What's the name of it?
The one that she likes is called a tuna cops.
Yeah.
Tuna cups, yeah. So they're tuna cups,
It looks the same.
and
They,
it's not your.
Walking across the seabed, it might be.
He's
Yeah.
gonna walk across the sea then.
That's his little legs there. His little
so-
fins, modified fins.
Hmm.
Yeah.
So it's not like your typical fish. This fish have modified legs, fins
Modified fins.
that basically moves like legs and it walks on the seafloor. So we are seeing this in action.
That's great. Let's just watch.
So
Isn't that great?
it's this is adorable.
Juliana
It's
has a stuffed toy.
Mhm.
Have you guys not seen it? There's a red one of these somewhere
Hmm.
around. Yeah, they're cool.
Oh,
Of course he's
I mean,
tanned.
just look at the
Orange is
structure.
fine, of course. The heels.
So those fins are like they use it to
Walker.
walk on the seafloor. Unlike other fish we see in the deep sea.
I mean,
And-
Jimbo, I'm gonna try to go around to his right to catch his face.
I'm good, thanks.
Yeah, it's something else.
And I mean, I'm no fish expert, but the structure in this,
Sure enough.
see some like,
Yeah, I'm gonna try to get to his side so you can see his face.
I mean, we are so amazed to see this species.
Sure enough. I'm butchering
True enough.
the name right now. Don't take what I say as correct. But it's something close to that.
Look, it's CHAUNA COPS.
Yeah,
Oh.
Sure
Trona
enough.
Cops.
Trona Cops.
Well, that's what somebody says. Take it with a grain of salt.
No, the viewers are normally right. I would believe what that
Yeah.
person says.
Let me just.
Yeah. So
Yeah,
we
that's-
just going
Trona
around
Cops.
Yeah.
Chona cups.
The genus of marine ravefin fish.
That's sea toad.
Sea
Hey,
toad.
look.
Good. So scientists and the ROV pilot guys are telling me that it's like a sea toad, and it's exactly how it looks. It has these modified fins that it uses to walk on the seafloor. So this little guy there is hiding his
In a dumb
face.
place, I can't-
Yeah.
He's smart.
Oh, So this guy's a little shy, but hopefully we see our other one.
He's turning.
Turning. Oh, okay. Yeah, introvert.
It's
Okay,
very rare to
he's
see them
gone.
fly though.
Uh-huh.
Yeah.
So you're getting
Do these
a good image of him, and he's definitely hiding from us.
Yeah. Okay, zoom in on him. You could pan and tilt on one and I'm flying very slow. I'm gonna go a little closer.
Oh wow,
There he is.
he is adorable.
He does look like stuff.
Okay, Jimbo, get all your
Yeah, I'm good, thank you.
stuff. Turn the laser off there.
Laser's off, yeah.
Zoom in some more.
Now he's staring at us. Oop. Oh, and he's okay. No, he's
Hold
good.
on.
Not a bad shot.
Yeah.
Great.
Oh. Yeah. Fish had a little clumsy moment. Oh, that was adorable. So that was the Chona cups. So hopefully we see other ones like these, or even different types.
So we were going to look at a coral when I first saw it.
Yeah. We can go. Yeah.
Yeah, you can resume.
Bridge,
What was it?
RV,
Yeah.
can resume.
We'll going to Point Tree. Yeah. So we're heading to Waypoint 3 and we're still bubble hunting. And we just had a nice quick commercial break, tuning into a Shiloh Chona cups. Oh, these corals are really nice. Like, probably zooming on this bigger one. Yeah, so we're starting to see some different types, seeing some yellow ones. The one below looks like a kind of ombre, white, yellow color. See some broken ones as well. Mm. These squat lobsters are known associates of corals. They just hang on these corals and absorb any or feed on any organic particles in the water column. See some shrimps. And look at this guy just hanging on the coral. And there's some other squat lobsters here as well.
I don't know if the guys did it. We could actually do it. They did. Decrease the ISO again. Close the iris over. Yeah. There we go.
So you're just getting a better view of the coral, and it's actually looking sort of pink, pale pink in color. Definitely different to the ones we have seen on this dive. Yeah.
Yeah.
And for those who have just joined us, we are at site 6 and we are bubble hunting. We are looking for seeps. So we are making our way to waypoint 3, and this is a huge crab. Yeah. Did, no?
Look at what?
Those corals up there, they seem to have something on them.
Oh yeah.
And all those white bits are-
Oh, the ones on the behind it?
Yeah.
Okay.
Yeah.
No worries. He's the king of the hill right now.
And this guy just sitting there.
Turn the laser off there and just get a picture of this guy.
Is that female, Anna?
Hey, Jimbo.
Yep.
King of the hill. Okay, moving over to
The
the
corals.
coral.
Did you say that's a feet?
I don't know.
Yeah, stop it.
So we're going to
Stop the vessel, please.
Oh, so this seems like it's just coral that is dead with some hope and stuff. That was the white part. Down.
so we have some corals here that are live, and it's also they also have some skeletons, some dead corals with it as well. So it's almost as though the dead skeleton provided a substrate for the living ones. And we're seeing 3 different species of corals here.
Oh, okay.
So-
This is interesting. Let's have a picture of this. It's like
Yeah.
a skeleton.
Yeah, it's a skeleton.
There's like 3 corals. Like the skeleton one, then there's the purpura, the master. All these are on skeleton.
So it's a skeleton
Mhm.
supporting 3 different species.
Yeah, so I think this.
So
one
what we're
here,
looking at here is the dead skeleton, which is the bigger one. So it's at the base. The skeleton is at the base, and then it's providing a substrate for the other corals. And we're seeing 4 different species here. We're seeing the pale pink one, which has the squat lobster on it. And we're seeing 4 different types here. So this is really, really interesting. The skeleton providing a space for the living corals. And seeing some small amphipods.
Right.
This is a good image here. Seeing a small bentictinophore as well.
Okay,
Yeah.
move on.
Yeah.
Give the crab a little spotlight again.
Yeah, why not? He sitting there waiting for
Close up.
us. Yeah.
Real spotlight moment.
Yeah. So what kind of crab do you think this is?
So I believe these are lithiod crabs. Oh. Yeah, so we're looking at a lithiod crab here. And these crabs have little spikes at the top.
No, no.
So you're seeing the mouth is most likely feeding on any organic particles in the water column. And so these lithiod crabs have a lot of spines on its top area, and they also have few spines on the legs as well. These crabs are very stunning. And so these antennas on these crustaceans help them to navigate throughout the deep sea. See small little amphipod. That's some beautiful shots. Yeah, I think we're good.
Maybe it doesn't feel like carrying the rights. Yet. On the flap underneath it, so I want to say it makes it
Mhm. Yeah.
straight look into that.
So we think this is a female because it has a protruding belly
Zoom out. Let me just
that could be carrying her eggs.
get a close view of her.
So yeah, it's likely that this is a female.
Zoom in as much
See,
as you want.
seeing that
Is that full zoom?
protruding stomach. Yeah, it's very cool. Yeah. So definitely does look like she's brooding her eggs,
Down a little bit.
and
Wow.
just the details.
Awesome.
Wow. I mean the colors and the surface texture. This is amazing. Wow. Definitely looks like the queen of the rock. So again, this is a littored crab, and due to the protruding stomach, it's likely that this is a female that's carrying her eggs. You could see in the frame here as well. So littored crabs are known for their
You can't really
spikes.
see any eggs, though. You can
Mm-mm.
see the hairs.
Yeah.
So she could generate, like, once they develop 40,000 eggs.
Yeah.
And what she does is she gets to the top of a rock. They're not ready. Like, what happens is that they start orange and then they will open up and well, as they develop. And like, get darker and darker. And when they're really ready to hatch, it goes up on top of a structure like this. It starts ventilating and releases them. So
Oh.
she's high up so that the eggs get taken by the current and transferred far.
You see,
Yeah, that's why they're
40,000.
too big, you know. I mean, she's not ready to do that. But she's practicing.
So
Alright.
these
So
little
moving
crabs are known to carry 40,000 eggs.
Moving on.
And when she's ready to, release her eggs, the eggs, they go with the current, and they can disperse as far as possible. So that was a beautiful shot of the lithoid crabs, and hopefully we come across more of these. And so we're going to continue our dive. Starting to see some more bacterial mats and some dead shells here. And some tube worms, some dead ones. Yeah. Oh, and so we have some living ones. And here's another type of crab. See, this one looks different from the last one. So we've seen some living mussels here, and you can tell that they're alive because of their color. You know, they don't have that white peelish color, and
Stop the muscle.
these shells, you can see those. If you look close, you could see like almost like a white flesh-like material. And so these are some signs that you can tell that the mussel is alive. So it's not a large mussel bed, but yeah, a mini seep.
Sure.
Mm-hmm. So you see now it's small, a small but mighty community. You've seen some tube worms. I've seen some live ones. Yeah, you see in the, I think.
People.
See now, a mini seep, seeing some mussels. So we seeing some live mussels, some live tube worms. These feather-like structures are used to trap met. I think we're seeing some bubbles too.
Where?
What?
Yeah, look in there.
Where?
No, that's not bubbles. What is that?
Don't scare us.
Oh no, but something is.
She's seeing like
Yeah, you see it?
thermal.
Oh,
Kinda-
I'm seeing
Oh.
it.
Yeah. What is that?
Okay, well that's
So,
it's other
Uh,
fresh water coming out of our bottom. No, it's flood.
Yeah.
fluid,
Oh,
yeah.
wow. Okay.
So we're seeing
That's very
some
cool. Very
some
good.
fluid.
Yeah. Wow. What an eye. Look
It's
at
seeing
that.
some fluid.
Yeah. It's up, look over here. By
Like-
the mussels too.
Yeah.
Oh, that's fantastic.
Wow.
So it's almost as though
So
these
definitely-
fluids are
So can we change the waypoint to also say?
coming from the mussels. So we're just
Small seep with,
With fluid.
with fluid. Seep, S-E-E-P.
so we see some bacterial mats, see some living mussels, tube worms, and these fluids that are coming directly from the mussels.
I know, that's what I'm telling you.
Nicely done.
Good job.
Wow,
Yeah.
what an eye.
So usually you see bubbles, but this time we're seeing fluid.
Yeah.
So
Coming right up.
we're just really taking in this moment here.
Look at it.
Do we want to take on the skin here?
A what?
Understand?
No.
Yeah.
Ah, no.
Because I think they wanted all
That's
6,
not
right?
what- So we have to fire all 6.
Okay, alright, never mind.
Yeah. And I don't think anybody's gonna analyze them, so.
Okay.
But what do you think is the fluid?
No, it's methane coming
So
out,
that's-
but it's like,
but
it's
that's in
just,
fluid.
yeah, it's,
Liquid
okay.
form.
It just
Ah.
doesn't come out, it percolates as opposed to bubbling,
Mhm.
instead of slowly coming out. Yeah.
And so these fluids are similar to the bubbles.
Check this out.
So
Look up.
usually when there is methane within the sediment,
Seeping out, see?
it basically builds up and it forms cracks
From underneath, that's the
and
seeping.
fissures with on the sediment surface, and then it escapes because of the pressure and the heat.
I mean, I've seen it at the
And when this happens, bubbles emerge. But this time we're seeing fluid as opposed to bubbles. So it is pretty cool.
It's pretty cool, though, right? Yeah. We have our temperature could we measure temperature, like, right where that
Sure.
fluid is? Like, maybe in between the muscles there where it's coming?
I think I might have to get a little closer. Let me
Oh.
reposition first.
Okay.
And
Get closer, get closer. Wow. Very neat.
So we're getting closer to take some temperature readings.
It's always
Um,
been muscles, so they're all gonna
curious to
go
see,
circling
you know, what that measurement is.
yeah, watch out a little bit.
around. So does
Should be.
again?
Still in it, yeah. Conductivity is in it. No?
Um,
Oh, sorry. Okay, let me porch in right there. Right.
stick energy. So probably
So,
Right where the fluid is, you know, where we saw that fluid? Basically.
so over.
Just click straight.
so right now it's reading 4.98. Now the temperature.
There we go,
reading,
yeah.
Right.
See
That's
that, right?
ambient, right?
Yeah.
Yeah,
that's it.
5.13. So ambient is about 5.13. Okay, looks like 5.13.
What's that? 5 point.
Ambient temperature is 5.13. We'll try to see what's in the muscles. Can you put it a little lower?
Conductivity for the 49. You want them lower now?
So come up and then kinda make it a little more vertical, I think.
Yeah.
Come up, bring it up,
Yeah, that's fine.
and turn your jaw right there.
I'll be working.
Okay, so nice and easy. We don't want to disturb the clams, right?
But in the pool, there's some activity.
So go to the right a little bit. Right, easy.
I don't trust that.
And then down, right there. Easy down, yeah. Easy down.
There is one in the
Bit more to the right. Down.
Yeah, you
A bit more forward a little bit.
Yeah, that.
A little more to the right and forward. Forward a little more. And go to the right a little bit.
See the 49 conductivity of building. 07.
Yeah, right there.
752 UTC.
Give it a moment.
Okay, freeze right there.
Freeze. 3.41 for the conductivity. I'm just not sure what those units are. 3.41. Yeah, I know what the units that they're conductivity are. I'll see if we can find out from.
It doesn't really.
Doesn't really seem to just
Yeah,
like
not a
it, so-
huge.
No. Okay.
5.27.
Well, it fluctuates between 498 and 5.27, so it's
Yeah.
like that.
This one is.
It's not much difference. Stabilizes it, so there's not much difference.
Okay.
And it's more like
Okay, bring your arm back. Easy. That's up. Up first. Yeah.
that's exciting.
Okay. So bring it back. You want to measure anywhere else or
No, I mean it's a very small area. That's the area.
portrait out?
Thank you.
So we just take some temperature readings of the fluid. So you usually see bubbles escaping from the sediment, but this time we see fluid. So this is
It's
pretty
saltier, right?
interesting.
That's why it's looking like that. It's saltier.
Saltier. Oh.
That's
So
why
that's why
it's,
it's
that's why
coming
we
up
get
in-
the slur. That's why you get that diffraction.
Ah, okay.
Because it's not hotter.
Mm-hmm.
So. Alright, let's take a look around. See if we have anything else that's interesting. Like some more patches.
So it's amazing what that mini seep gave us. Seeing a lot of dead
Wow.
tube booms.
There it is.
Oh
Look at
yeah,
that. Wow,
yeah.
nice. Let's just stay here for a moment. Yeah, nice.
Good eye.
You have the good eye. You put us on test.
You put us under. That's it.
Fluid or just temperature?
We don't think it's temperature
It's
because
not.
it didn't really change the probe reading. So
It has, it's a different, it's probably more different, what do you call it?
Salinity.
Salinity, like conductivity, right?
Yeah,
So
so
that's why I'm
you're getting
wondering
more of a picnicline than a thermocline happening there. Just
Yeah.
over that little area. The density change, you know, so
Yeah.
much more dense water than the estate. So.
We wanted to find out what the conductivity meter is.
There it is, yeah. Yeah, I went up a bit.
so you can see it's coming out from here and then that's where it's
Yeah. Wow. So again, some more close-up images of the fluid that is escaping from the seafloor. Yeah. And so this is you can tell this is a an interesting energy source here. And so at this site, we took some temperature measurements, we took some conductivity measurements as well. And in this area, it's also said to be
Now we'll look
have a
lower,
higher
you can see where
salinity.
it's actually coming out from the seafloor. Yeah.
And so that higher salinity is basically is causing it to have a fluid like appearance as opposed to bubbles. And I'm sure there are other reasons why, but you know, this is really, it's really interesting to see.
We had that coming out and then mirroring like the it was just the Lux. We had 3 months.
Wow. Very cool.
So it's a lot.
Okay. There's bigger things out there, folks.
Alright. So we can continue.
But it was the
on
change.
that.
Okay.
Yeah.
I think we
Alright.
could keep
Someone got it.
somebody else here. No, you're on your own. Nobody. It's just us. I don't know who's next. I can check.
Crabs be hiding behind.
Sure.
Yeah. Okay.
All right, perfect.
Moving on.
Moving on to waypoint 3.
Yeah.
Thank you.
So we're continuing to waypoint 3, and we've seen a lot of. We had a lot of great commercial breaks on our way to waypoint 3. Saw little crabs. And we saw this fluid escaping from the seafloor. And so
So we
From
have a
this mini
waypoint
seep.
there, correct?
Yeah, two
Perfect.
worm, no, mussels, small seep fluid.
Perfect.
Lazy vibes.
So that small seep, small but mighty, you know, it's really different from the others.
Let me catch up with you.
Oh no, you're right. It's you.
Okay, so that's it for me, guys. I have completed my shift, but it was really nice chatting with you guys. We've seen so many unique and interesting features. We see a coral skeleton providing a substrate for living corals. We see a lithiod crab potentially brooding her eggs. And we have seen these small mussel seep that adds fluid escaping. So I'll now pass you over to my colleague, Robert Cassidy. And good night and good morning to, oh, and we also saw some Chronocup as well. So I will now pass you over to my colleague. Good night and good evening, good morning to wherever you are. Thanks, guys.
It's a unique seep. It's fluid coming out as opposed to gas.
Look at all those
Right,
tubes.
mussel, small seep fluid. That's the name of it. So it looks like, you know, like when you're driving on a hot road and you see it like the heat. It will look like that. You zoom in. Yeah.
Put it inside and come
Alright,
in.
so we're going towards waypoint 3, but If you see anything interesting along the way, you just
Okay, this is
look at it.
look around.
Yeah. But it's really rocky and stuff, right? So just mind if you come down low, if you're turning,
Yeah,
just kind
there's
of
muscles here.
Yeah. And that heat probe, it's was installed fluid. The heat
So this is definitely seeps, right? Can we?
This didn't chance.
Can we stop moving while you're talking?
What?
Can we stop moving while you're talking? Because one time.
Okay.
Yeah, thanks.
Because it's not going. It wasn't.
This is different.
Hi everyone, Robert Cassidy here, taking over for Eugenia. I'm going to continue exploring this very cool seep area that we've been looking at.
So instead, yeah, it should be there.
Yeah, we need to be a little lower.
Because she said, yeah, well, when I was doing it, I was keep it there. But she said like 3 sensors was penetrating the stick. So I think you need to just when it comes on there, just see. She was saying, Rachel was saying that. Like it was really nice. I think it was just we didn't. I don't know. We didn't use
Nice view.
I'm just bringing it back up so I'm the little all good.
Yeah. I mean, I know we've been looking for mussels, but Tiburon's living seeps too, so I don't know where we always say this is not. But this is a massive Tiburon area, so it'd be nice to get a good overview if we can get close to see if there's any more fluid coming out. It's big. It's like it just
Okay.
this is the end. It's on this side.
Okay, so we should kind of go that way.
Just where we came from. Just going. Just look around this whole area, but more to that side.
Okay, sounds good.
And let's zoom into the south episode. If we can, a few Tiburon bushes to see if there's anything coming out.
Okay. You want a waypoint here too?
We will put a waypoint in, yeah.
Okay.
But I want to see it first. We haven't, because
Yeah.
they were flying in and then it's fished.
Okay. So we want to get a better look at these two worms and the seep area here. So
We do.
we can zoom in here a bit, and we want to kind of also look around to the right after that.
Sediment if possible, like,
The sediment, like so down more.
yeah.
So yeah, we're looking at the sediment mostly. So got a lot of live tube worms here. Well, they're live all over the place, really.
There's
And
a
it looks
lot of
like
them.
there's a lot of them, and it looks like is that two types.
Yeah. So the long,
Yeah.
so the curly ones are one species.
Mm-hmm.
And the straight ones, the long ones we see,
Yeah.
is a different species.
Yeah. Wow.
And then you have the superiors, which are yet another thing.
Yeah.
There's actually some huge bushes over to our right.
Okay. Great.
See what's going on over there? There's a lot of stuff.
To the right.
Yeah, well, all this. So if we could just
Can we pan to the right, please, and maybe focus on
maybe fly over it. But
this area over here?
see what like over massive bushes.
Yeah. Wow, look at these huge clusters of tube worms.
And mussels, a few
Mussels. Yeah, wow. Maybe we could zoom on this area a bit on these rocks. Wow.
Specific point for
So,
Yeah.
the Scarpia require, these guys don't
Mhm.
need fluid, but the other
Okay.
ones do. So if we're gonna zoom anywhere, we should be looking like,
At those.
yeah,
Thicker
so we're
tubes, right?
over a tuber bush right now.
Okay.
So if we
Can
could
we
just
back
sort
up
of-
a bit? Okay.
It's under our belly.
Sure. Yeah. So yeah, we want to look right down if we can, because apparently
Yeah,
we're
see,
over
there
one.
it is. There, to our, just,
that one. Okay.
This one, or is it more over
there,
here?
see those ones?
Yeah. Yeah. If we could just back up a tiny bit and look here, it's great. Got two mussels right in there too.
Keep going, or if you want to see something on this bush.
Well, we just if you could just zoom out a bit and just fly over, because what happened is we came in as this area as you guys were shifting, and so we didn't actually get a good look at it.
Okay.
So if you could just sort of backtrack a little bit, we can look at this overview, and that way.
What's that?
It's a lot of stuff.
Yeah.
We're gonna need a face because there's so many rocks.
Okay.
Which side they're gonna hit. So.
Yeah, so this tuberums is also some on this mud, so they're really
Okay.
underneath us.
Yeah. Maybe we could go up a bit and just back up to Boyhaven. And then we could zoom in, come back down.
That looks good.
Yeah, that's good.
Yeah. Zoom in. Yeah, that's great.
Yeah.
So if I could put a waypoint here called tuberums, please. Tuberum bushes, actually.
You go over.
So if I could, zoom out a little bit because it's hard to focus with. We're just moving too much and it's hard to, well, not that, a little bit less. I'm trying to get a good look, but without all the movement.
Tubo worm, garden, or what was?
Bushes, bushes.
Yeah.
Is it possible to, oh, I guess yours.
What one you want to look? The one on the right or the left?
Both. The one on the left, probably. Sorry.
Yeah.
The one on the
one
left,
first
please.
to look at.
Yeah. So left first.
Yeah,
Good.
that one. See, here we have some muscles in the one we're passing on now as well. I mean, this side below us is pretty flat, isn't it?
Oh.
This side below us is pretty flat. So if you could go back a bit, and I don't know if you can sit down and we can take a look at those two bushes. Those two there.
I think if we perceive both and it's sit on that direction.
One of the two muscles is the one up.
Okay.
The only place I can sit is here, because the other side has another.
Yeah, that's fine.
Yeah.
That'll be good because then we'll be ahead of us, right? And a bit. Yeah.
Yeah. Yeah, that's great. So we just want to zoom on this one here for a sec with the muscle in it.
Straight ahead.
Sit down
That's great. A little bit, yeah. Wow. So you can really see these two different types of tube worms right here. Very different casings on the tubes and also different kind of feathery heads here.
So basically the purple ones are the white ones.
Yep.
Can we pan up a bit? please?
Exactly. Yeah, can we go up a little bit? Thanks.
Yeah. A bit more.
Maybe
So there'll be a
around
couple
that
of
muscle?
muscles. Yeah, there it is. Yeah.
Yeah. If you could maybe put that muscle like around the center, I'd say. The muscle in the top right corner, you could kind of center the, yeah. That's good, yeah. Yeah, I'd say that's good. And maybe we could keep going up a little bit too. I think there's another cluster up there, right? Yeah. Yeah, so we want to try to see that, yeah, this other cluster. You could, yeah, that's good. Wow. Very cool.
Can we zoom out a little bit so we get a bigger.
There's a scale worm in there. Too, pretty big one it looks like. Right?
Oh yeah.
Yeah.
It is.
Wow. Yeah. So,
Perhaps.
is there any way to zoom more? Or is that the max? Okay. No worries. Yeah.
More than this? Yeah, that's what that costs. Microscope.
Right. Magnifying glass. Yeah, so this pink scale worm here is kind of similar to ones that we've seen before at seeps on this trip. We have some new DNA data we got a few days ago that looks like it might be a new species. It's very curious to see what comes of that when we do more taxonomy workshops and things like that. Okay, do you want to...
Yeah, zoom again. I mean, focus.
Got a squat lobster on the left as well.
I'll top it off.
Very cool.
Very nice.
What do you think? Should we keep looking around, or you want to
Yeah.
stay here
No,
more?
I think we can keep looking around. Yeah,
Okay.
it's great.
Yeah.
Thank
Thanks,
you.
guys. So we can just keep looking around this area, but now, you ready?
A lot of tulips here.
Yeah, huge amount.
It's another one there. It's just, you haven't seen this before.
Yeah.
This is what the,
So do you know why there would be so many tube worms but no mussels here?
I don't know what the mechanics here are, but like in the Gulf of Mexico, there's some that are dominated by tumors of seeps and some that are dominated by
Okay.
mussels. This is sedimented, so
What do you want to go to here? In what direction?
What do you think, Anna?
That wall?
It seems like we've come from the right, so should we go to
we've
the
come
left?
from the right, so we should go to the left and up.
Okay, yeah, so to the left and up is good.
Moving laterally to all those rocks.
Sorry?
You don't want to go up to the top of the mountain. Just move laterally.
Well, just to cover this area and then we go
Yeah.
up. Yeah.
Exactly. So we'll just scan around here, basically, and then we'll go up. But it looks like the worms kind of end here, don't they?
So we can go up.
Yeah. Alright, yeah, I think we can go up now and see it's in that direction, maybe. Yeah, we can go up.
Hill.
Yep,
Good hill.
thanks. Some more tube worms over here. You can really see this white bacterial mat as well. These are all just very clear signs of seep site. Oh yeah, mussels here. The kind of crevice in the bottom right.
The butterfly.
Oh yeah.
Hidden between the rocks.
Yeah. Is that Looks like a big claw from a crab on the ground. Oh, sorry, a clamshell. Cool. I guess we could head up this way a bit, maybe? Going up? Yep. Yeah, some more just really dense aggregations of tube worms on these rocks. Wow, just very thickly covered. Yeah, well, I see, well, not much right now.
Okay.
Just keep going up there? Yeah. So we just keep going forward.
Let's head towards the waypoint, I guess.
Towards the waypoint? Okay.
We can turn a bit.
Okay.
Well, just to see what's. Let's just turn there a bit to the right, to the left, so we can see what in the sun.
Okay.
This angle on or it wants me more into the waypoint?
Well, I mean, who knows, right?
Just kind of
let's go
seeing
like
what we
this
see.
for
I
a
guess.
while, and then we'll see. Yeah, who knows? I think the reason they put the waypoint there is because they want to cross that rim. There's a rim.
Yeah, right.
So you see at the top, so they want to go across.
Okay.
So that's why I think it's there. But basically, we look at the seeps. So if we find them, we stop.
We'll look at the moors.
More seeps, I know. I think those two worms are fantastic, and I think
Yeah.
we're sort of.
Well, you have the tube worms, you
I
have
know.
mussels.
Yeah, the other one. Well, the other one we actually had seepage.
We did see the occasional broken clamshells, no line on it.
Yeah. So I know we're looking for mussels for some reason, but I mean tube worms are, especially those decis, there's evidence of seeps.
I thought mussels were one of the indicated species.
They are. That's tube worms, but for some reason we're only focused on mussels. But we just passed this huge bed of tube worms. Tube worms cannot survive without the mollusks can. They quick feel if they feed like mussels do. They're not as good at it as mussels that don't live in these environments, but they can. Whereas tube worms cannot. They have no choice.
What is that? Could you zoom on this real quick on this rock? What are they on though? Is it? Hmm. That's good. Thanks.
Going up.
Yep. Still hadn't.
Okay, once a minute you'll get a seep, don't find anything.
Okay.
If you do, stop. Have my phone. I'll scream through my phone.
Charlie. Hello. Could you move to this updated waypoint, please?
Could we just fly by up there by the white area? Yeah. Some more tube rooms right here. What's that? Oh, I wasn't. I don't know if that's sponge. Might be encrusting sponge. Oh, I see that one. Could you zoom in on yellow too? Yeah, right there. Yeah, I think that's good.
Bigger rocks right in front of
Oh yeah, we can back up a bit. Yeah, we could go a bit and head more towards the waypoint now, I'd say.
More left.
Yeah, just a little bit more left. I mean, kind of, yeah, point that way a bit.
That thing there inside.
Oh, whoa. Look at that. It's massive coral, looks like. Oh, it's dead. Yeah. Yeah, if we could take a look at that, yeah.
What one on the left or this one?
The left. You zoom it a little bit. Yeah. Yeah. Huge bamboo coral. Can't tell if they're off or if it's out of frame. They're on? Yeah. Yeah, so we could get another zoomed out with the lasers for a second. You can see them. Yeah, we just don't want to get too close though.
You see the lasers in the picture there, is it here? You see the lasers?
No.
So we could back up a bit maybe, and I don't see the lasers anywhere.
No, yeah.
Oh, I see. I see where they are. Could we point them towards the coral more?
Want them on the coral, so that we can measure it.
Yeah. So maybe if you like pointed the lasers at the base a little bit, it'd be easier. Yeah, we don't want to get too close though. Can you back up at all? Yeah, I think that's good.
sit here?
Hmm?
You want to just sit down?
No, it's okay. We can go up and keep going. Thank you. To the right. Yeah, can we check out what's over here?
Oh, it's that stuff. No, that's
Okay, that's good. Faster. Okay. Yeah, we can move a little bit faster if that works as we go to waypoint 3. Something in the distance over here. If we could just kind of arc towards it. Looks, it's just like white mats, looks like. Yeah, that's good. You don't have to go too close. Yeah, we don't need to stay here really. If we could just kind of move a little bit quickly towards 3, that'd be good.
Okay, I'll leave you to it for a little while.
Yes. Then freeze it.
Yep.
So the switch. When you switch it towards you. Whichever one is quickly on when the alarm's coming up. That's away from the driver. Is this way. Keep it towards you. So if it's on now, it's not running. It's buttons cut. It won't run. That way you see how it's up there.
Do you want to stop this squirrel now?
Yep. Was it audio alarming when you left it? It's probably come on now. Yeah. Just silence. Double tap.
Double tap will try.
Okay, double tap and hold it and then it does stop.
I tried holding it, but
Put it over the side, whatever.
well, that was weird. It went off by itself. So.
Looks like another resinged star here. Yeah, we don't have to stop. We can just keep going.
Like to make some ground.
meters ahead is different too.
Yeah. So I think do we want to go right on waypoint 3, or do you want to
Cut the corner?
cut the corner? Up to you. I mean, it said to explore it's like the local high, but I don't know what else.
Yeah, let's just go to the
Yeah.
local high and
Okay. So yeah, I'll just.
use the local high. Now the local high may have less sedimentation and we might want to fire the
Yeah.
Niskins.
Okay.
I mean the other option, I don't know the deal with the Niskins. We know that Laguna is not going to work. So I'm just thinking we can do it as last because if we find a spot,
Yeah.
we want Niskins, then we'll just fire them all. But at least
Yeah.
we can do some of that water.
Yeah, I think that makes sense.
And if we don't, they have to fire them again.
Yeah. It wasn't clear to me if Ladana wanted to be there to fire them
No.
or no. She just wants to
She
practice it.
doesn't care. She just wants to see.
How getting the water out all works
Yeah.
and stuff. Okay.
Thanks, Dmitry.
How much water and all this.
Sorry?
We are on 3.
Oh, we're on 3.
All right.
We
Here we are.
do a
Top of
little-
the world.
Yeah. Top of the world. I think we just kind of scan around for a sec. I don't see that much here, but
Yeah. I think we
just go to 4. Okay. Yeah, we can just go to 4 actually.
The next one.
Yep. Thank you.
just go for
Just rocks.
Great, Gerald. Do you please update a waypoint so you can send over?
Are the lasers on?
Yeah.
Oh, yeah, I see.
It's pretty rocky here.
We go to the highest point or keep falling laterally like
Well, if I found it follow the lead.
Yeah, that's a good idea. High point, and then we can follow the ridge as we go. Thanks.
for me it's easier to fly.
Perfect.
I don't see what's going
Right. Yeah, yeah. No, it seems hard to fly on the slip.
You can now see what's going on from out there.
Yep. Win-win. big rock here. Lots of rocks. Yep. Yeah. A lot of nice corals over here. Black coral, some squats right in front of us. Some big ones. Over there. Yeah. Wow. We don't need to stay here though, really. Do you want to go zoom?
Yeah, we could do
Zoom on now? Okay. Yeah, can we zoom on this, please? This is a black coral. I've been seeing some pretty big squat lobsters at some of these sites the last few days.
another egg? Skate egg?
You think it's an egg? I was just wondering if that's an egg.
Skate egg maybe? Or yeah,
It-
let's take a good
Yeah. If you can just zoom on this right here.
Put the rims out and park
make
against the
it-
road.
Sure. Yeah. That'd be good. So we're seeing what we think might be an egg on this black coral. Gonna get a closer look. We've seen some octopus eggs on corals the last 5 days or so. This one looks a little bit different though. So maybe it's a skate egg, like Anna suggested. Yeah. Great. Yeah, so we want to try to get a zoom on this kind of egg thing here. Oh, wow. Oh, great. Think so?
Looks like
Wow. So yeah, it looks like a skate egg. That is cool. And it's a beautiful color of this coral also.
just the whole thing is just beautiful.
Yeah, it's gorgeous. Gorgeous.
Yeah, a little sharper on the end.
Nice. You good? You want more? Good. That's good. So I think we can probably get one more quick just pause on this coral here. That's good. And yeah, then I think we're good now. Oh, okay. Very nice. Yeah, I think we're good to move on. Thank you. Okay, so I'm gonna pass you all over to Charisse. I'm gonna keep exploring this cool area.
Morning, everyone.
Thanks, guys.
Hi, good morning. I'm good. Alright, so I think we so we're just gonna push back a bit and then yeah. So we're just trying to when we push back we can get close and head around to good morning, everyone who's joining us right now. It is 5 a.m. local time as we continue exploring this location. Seems like I've come in when we've been watching some pretty corals, so that's exciting. And we're just going to continue along a dive path in search of additional corals and sponges. It seems to be a bit of a rocky outcrop area, which is great for have hard substrate for things to stick on and live on. Which is usually when you start getting those corals around. Seeing a lot of shells and tuberums coming up, which are indicative of seep areas. And on this dive, we are indeed looking for signs of seepage and bubbles. So these are always good signs to come across because it means seep activities within the area.
crab in the corner of the screen. Do we want a quick zoom of it? Or pictures or not? The crab?
Yeah, why not?
Can we get a quick zoom of the crab bit? Nice. Nice. Yeah,
sorry.
we can continue with that. Thank you. Yep. I see two berms, but not really. No mussels. Shrimp. Yeah, so we were just discussing whether we're seeing tube worms. They are superlids, so they're not entirely tube worms. But again, good indications of potential sea activity within the area. And I don't know if you have the map on your screen right now, but I think it was shared earlier. It essentially shows the track line that we're taking, and those red points are potential bubble sites. So when we get to areas like that, we just take a circle around to see if we can find any like signs or activity. There's a benthic down there. A lot of shells down there. Yeah, you can keep going. I don't think anything's really alive.
Need shrimp swimming along. Looks like a black coral with some squat lobsters on it, potentially, as well. Yeah. Did you want to zoom a bit or you're okay? Yeah, we keep going, yeah. That is a mound of shells. What? Wow. There's some mushroom? Yeah, no problem. Yeah. No, we're good to go. We're trying to make it to point 4 already, where the bubbles out, but if I do see something, to slow down.
0.4.
Yeah, basically. But if I do see something, I will tell you, and we can maybe stop and take a closer look. But that is a massive amount of shells. It's
There's a lot of dead shells.
yeah.
Wow. Wow.
Whoa.
We're live at the duck.
Yeah, I was like, we're coming into the live shells right now.
Okay, here we go.
And this is when we keep a lookout for bubbles, guys, especially.
So where are you seeing fluid?
Yeah.
Yeah,
Then we're at an active seep because look at all those live mussels.
now we can slow down.
Now we can slow down as we examine this for a bit. That is gorgeous.
It seems like it's a mix of dead and live mussels.
Yeah,
so
and it's almost in a line.
Follow the line.
No, I think we continue along to waypoint 4.
Well, let's just see it, and it's pretty big, so let's just fly on.
Fly
and
up
see
and
how
okay.
It's pretty massive.
We could circle around it in here, fly up as we follow along with the living.
But can we have a waypoint, please? Mussel bed.
Yeah.
It is huge. Yeah, it's huge. So we could just explore here a little bit.
There's a crab up there.
So with us.
Yeah, we're just gonna explore around within it.
What you actually want to know, tell me what you want.
Should we go a little bit up in like a circle?
Yeah, just do like a circle around
Yeah.
it just to see how big this thing is.
Yeah. So we're just gonna circle around it and just get a general gauge on the size, because it seems massive. Are these childresses? A gigantuous. Yeah, we saw it earlier. Thank you.
Yeah, it's massive.
Whoa.
I think it's the biggest we've seen.
Yeah, I was gonna say, I think this might be the biggest one we've seen.
I think we'll spend some time here. Just we probably want some samples. Just want to figure out like.
Oh, there's an eel
Extent
pout
of it.
swimming in the bottom right of your screen right now. All right, guys, we're all pretty excited over here because this seems to be one of the bigger mussel beds in the seabed area that we've seen so far. And we're just taking a general look all the way around just to see, get a gauge of like how big this really is. We've also put down a marker on the map just so we have it, we know where the site was along our transects, our waypoints essentially. Whoa.
Wow. Huh?
Still good.
Yeah.
Well, if that doesn't wake you up at 5 a.m., I don't know what will.
For once, it's me and not Beats.
It just
Wow,
keeps
that's
going.
Massive.
Yeah. We're seeing two burns over there. Eel pout swimming through. This is stunning.
Keep going around.
Have we made it to about what looks like an end? Maybe go up and see how far
Yeah,
up
just
it
need to
extends.
Map it a bit. Yeah, no idea. Well, what are those? Are those serpulids? What are all
Are these? Yeah, we're gonna
Or
head
are
up.
they small?
Depends
Oh.
where we see it, but it's so big we can shoot in another place for you. It's huge.
Yeah,
Can we
because we're
zoom
gonna
in?
head up and around. So
And go
can
back
we
down.
come back down and like zoom in
Yeah,
on this
trying
area?
to figure out what they are.
That's no problem. We're
Oh,
gonna
those are
see what
serpulids. They're serpulids. I saw them. So
Oh,
they're the
serpulids.
ones
Okay, so we don't have to get down. But
What direction want you to keep circling?
maybe
Across
head
or
up
down?
and then
Across
yeah,
over.
and
And then come back down.
come.
Just sort of get an area. An idea
Yeah,
of the area.
we're still trying to gauge how big this is.
Oh, looks huge.
What's David's room number? Anybody remembers?
Oh, I have no
Are
idea.
those- Is it?
It's in the lab.
I don't want to call somebody else.
I can check it.
Sure.
Whoa.
Wow.
We can stay up, but yeah. Look at that crab. Yeah. I think we've seen about 3 or 4 crabs around here already. Yeah, we can keep going over.
Mike, how big would you say this is?
The whole area.
Yeah.
Well,
Let's look at this in the
oh,
back
look,
too.
I start.
1561.
Guys, we're not muted, man. Still live.
1561?
Yeah.
We're talking about the area. We start more or
Oh.
less comes around here.
Yeah, we
It
can.
just keeps going.
We were seeing from here more or less. That's bad.
It is a massive.
Yeah, I think we can.
Here,
You haven't
look,
seen
here is 20, at least to this edge, 30 meters long.
Okay, David's coming
Yeah.
down.
Okay.
35.
Yeah, we can go back down and just keep exploring along.
35 from the line on
Yeah.
this side.
Oh, nice.
Yeah, it'd be good if we could just, so once we get a sense of it, just get a flyover, just like this thing. Wow.
I start to come right here on it. I start comes around.
It comes
Yeah.
around,
Here.
eh?
Can
Yeah,
you
but
see my mark?
sorry.
Look my
Mm.
hand.
Yeah.
There is where she asked me to start. Comes around.
Okay.
I saw and I start to do that sideways.
Yeah.
That's
Yeah.
why we are looking here. That
Yes.
means
But
the
then-
whole area there maybe even on the other
I
side.
mean, and then if you look, actually, if you look farther, see, there's more over there.
Yeah.
Need to cross over.
Well, let's just
We
wait.
could
Yeah,
go-
we can, and then come back. Sure. Yeah. Let's go across this side now, like on that side.
Okay, following the edge here.
Yeah, exactly. Let's just follow it.
It's at least 100 meters long.
It's huge.
Wow.
Like we haven't seen anything like this. Is like, well, for here for sure. All our dives we haven't.
Yeah, I'm following along the edge to go that way.
Okay, so we see the end over on that side.
Yeah.
Right?
Almost back of the line.
Do we wanna maybe?
Following.
Yeah.
You in between. You want to see the line or you want to cross?
Let's go so we get all of it within the stream itself, if we can.
So, did we come from there, or is that carrying on and then?
That's
Oh,
carrying on.
You came from here.
Right. Wow. Wow.
That's
Yeah.
not bad. Try to make it bigger for you to see why it's already more bad.
Yeah. There it
Came
is.
here, then we start to lift around to see
So-
how big it was.
So right there, we still had. That's when we first saw
Yeah.
it before we turned, right?
We
So
saw
we-
more or less here.
Yeah.
Then we come. Then he told me, oh, start goes around.
Yeah.
Then we came all the way here.
Yeah.
It's big.
No. Yeah. It's wow.
It's massive. And look where I am now.
And yeah.
I'm not even on the line yet.
I know. So I think we're gonna stick around here just to sort of, well, first of all,
But
Do we still
Diva
wanna
is on her way. So she
continue
needs to
like
see
going
this.
along the edges to potentially almost map
Yeah,
it
sure.
entirely?
Yeah, we can sort of try
Okay.
that. Yeah.
Yeah, so we
Yeah.
can do a perimeter on the entire patch, and then we'll almost see
okay,
the track of
follow
it.
the border, the white
Yes.
and black.
Let's see what we see. But
Do we
this is
wanna
now on
follow?
this side, right?
Yeah.
So we've sort of reached that end. It looks like.
There is another one.
I know.
Maybe we drop a bit and
Yeah.
see what
What's
is
on
there.
that side?
to
Sure.
see if
Let's
it
go a little
starts.
bit. Yeah. Although there's a lot of dead mussels here, so I find it.
That's the end. 'Cause
Yeah.
we start seeing, you have the marks.
Okay. Right. That's the end. All right. That's great. So we need to turn.
Turn
Right.
and
Keep
yeah,
going
following
that
that way.
Follow the
The edge on
Yeah.
this end, and keep the camera
the other
looking.
side.
Follow the lines, just so I see where.
Can okay.
Yes, this is edgy. We see mixed muscles.
Yeah.
So we need to turn. Oh.
Can we turn to really keep the live muscles within our baby? Yeah.
Yeah.
I think we may have just found the end.
Yeah. So let's go.
And
And
go
map.
to the edge.
I think we could just turn at that point and just continue back along into the massive live bed.
Yes. Yeah, that's right. Right. Turn right.
Turn right, yeah.
There's sponges here, we see. And there's bacteria. bunches.
Yeah, I think that's the edge as well there too. So we can take a right again and
Yeah,
just
just go
go
right
back
again.
down.
Oh, you don't want to see this thing.
Yeah. It's a pretty nice patch as well, but it's ending. So I think we just follow back into the massive bed.
Okay, so can you zoom out so I can show Deva the extent? Your
Okay.
look. This is what it looks like, but look to the. So we started seeing them where the line is. Show, yeah, maybe you can show them.
Where
Not
is my
there.
hand?
Yeah, where.
More
so-
or less here, then we start comes
So that
around.
whole area that you see where we are, the whole area. Okay.
Okay, yeah. So we're going to turn
So we're just
back
doing the
around and then basically head to that whole area that we were in just now.
We saw the big one.
Yeah.
The massive volume.
Yeah, that whole big section. So we're just gonna drive over it. And potentially there might be sampling happening. Not entirely sure yet. But we just wanna.
So
Can we turn a bit right again? And then
we can get a little closer.
closer.
A little closer.
A little.
And then,
I think.
yeah, and
Here
then
is
if we
okay?
could go back to just sort of, because Diva just got there, so
Yeah.
she needs to get a sense of the size.
Mm-hmm. It's gorgeous. Yeah, so essentially what we just did is we tried to follow along the edge of the mussel bed to get a gauge's size. It almost looked like 100 meters long from what we were seeing. And now we're just going to head back along and really take a closer look at it. Sorry?
Can we make a circle?
You're right on the
Yeah,
top.
we're just going to keep following on
So
the,
it's kind of
along the
right,
hill.
so it's kind of sloping up and then, so it's kind of undulating. Just right.
Can we angle a bit more right as well so we can keep the healthy, the alive bed within view?
Yeah.
Perfect. Thank you.
So where did I mark?
I think we're coming up to the beginning mark. Put in. Are the lighter colors in the younger ones? Younger muscles?
Yes, the lighter color ones. Yeah.
Wow.
The last night we made the flag with heaven, all of these points, but they weren't.
Yeah, yeah.
Or they're
I was
not.
there. I know.
They're not.
So
Just gonna. So I would actually consider like either running perimeter or running mosaic.
I
Running
know.
mosaic,
Yeah.
but like.
So we try to get the perimeter.
That can be the only place that we used
Sorry?
I think I like it between the
Yeah, but what we'll continue doing is essentially probably follow that same path that we did earlier. So Diva sees it as well.
There's lots of hot.
Yeah. So yeah, exactly. Yeah. So what you guys are seeing in your
So we could be back on the edge, So where can we go back?
Yeah, we're gonna follow that exact
Okay.
line back down as well.
And then sort of get to the
Really
thick
thick.
area.
That's
Yeah.
where we started.
This is where we started.
Yeah,
The
so
only
we're just
difference I did sideways.
Yeah.
We want me to repeat on the same position? Because then I was looking.
So I'd like
I was
Diva to
moving
see
like-
the extent,
Okay.
and then
I
once
can
we've
fly
done that,
over.
then we're gonna have to be a bit more organized about getting an actual perimeter, and then doing some transects through it.
Okay.
But once we get a good idea, but.
So is
we
it
could
okay
run
to
it
follow
as-
that same essential
So you know when we first found it, there was all those tuberums and not tuberums, superiors, right? at the
Yeah,
edge.
it might be
Yeah.
us turning into that position again. Because it seems like we're basically there.
On the top, it's not long again.
Yeah, we're right on the top of.
But where
I
are
wonder if
the?
that's what that. I guess that's what the sonar is showing. Yeah. You mean the first time?
Like well, I don't know how many times we've seen seepage.
Here, never. We haven't seen it because we fly high to see the. We haven't seen this.
Oh, when did you see seepage before?
There was in another place.
Is that
There's
what it's gonna be?
like four muscles, thick muscles, that fluid is actually coming out. So it was actually two seats.
Quero ser para mostrar para ela.
I think we got that a little bit.
Okay, so
Do we want to get back onto that one?
so what we want to do
Okay.
is this is the edge.
Yeah.
Like we're seeing an edge, so
Yeah.
that's great. So what we need to do is we need to fly. We need to get a measurement
Okay.
of what this is exactly. So we need to grab the edge
Mm-hmm.
and do the entire perimeter, like complete, because now
Okay.
we've been going in and out.
Mm-hmm.
Just follow that edge and get the full size of it as
Okay.
a first step.
Do we want to put a point when we start and
Yeah.
end? Okay.
Absolutely. And we should be dropping points
So
along
we've
the
been
way.
doing that over here. Leo's
Ah.
been putting points as soon as we could get each of the edge. The more edges we get, we'll obviously fill it up.
Yeah, but then there's sort of a strange path there. So I don't know what that edge is. But what happens when we go in and that's a nice, but then,
This edge.
well, I guess we need them more frequently because I see a lot of turns with the RV and I don't know what that was the edge. You know what I mean? So that is my
We'll just do it more complete. is that-
systematic.
Okay.
Yeah. So you see it like here, like what happened here? Is the edge,
This
is
is
the
what.
edge.
This was like this. When we reach that point, there was from there, no more.
Okay, but what about here? Like how far did that go? You went flying along the
Yeah, we were kind of close. We were able to see. Remember when we were flying over?
Yeah, yeah. But then here again we're flying through muscles, so it would be nice if you just hug the edge all the way. Because this was not. So here, is that, I don't know where the edge is on this side. We just want so, yeah, so where we need is we need to do sort of a
So we'll do.
systematic, and then you can drop the spots, and then we'll know that that line is.
But for that, we need to be on the edge of it.
Yeah, that's right. Yeah. So basically that's, yeah, if we could follow that edge, then we could get a sense of, like a
Do you want to?
map of it.
You'll see this nail trail.
Okay, so this is, let's say this is the healthy about on the setting, this is the edge. Do we want to position ourselves this way, keeping the bed in view at all times and then going around, that's it?
I don't care what we do, I just, yeah,
Just follow
probably
the
that's better, but
Okay.
we need to be right
On
know,
the edge,
edge.
and then he will plug in the planes.
Yeah,
across.
as we go along.
Okay, no
Is
problem.
that okay?
Yeah, sounds good. Thank you. All right, Mike. So essentially what we want to do is we're going to get onto the edge here, and we're just going to continue along the edge, trying
So
to
we're
keep
gonna
the
go so
muscle
use-
bed itself in
Yeah.
view. And we're going to hug that edge the entire time. But we will continuously drop points as we're going, so we can properly mark out the outline of what the muscle bed looks like.
Okay, follow, you want to start in the following that direction? But that's uphill.
Which one is easiest with the ROV?
On this kind of terrain, I don't see rocks sideways anyways.
Okay.
Let me
So I say we get on the edge and just follow.
left.
Yeah. And we want to stay.
So we have to just go ahead and like, oh, you cleared it, perfect. And then we'll have it.
Can we? Okay. Let me.
We're going to the left, right?
Okay. Quick question.
Well, we haven't really been doing very much, but yes. Yeah.
Okay. If we could actually just position ourselves exactly in it and follow up, that would be great. Perfect. So essentially every point will be directly in the middle of where we are. Yeah. And just follow along. First point done?
No, clearing mine.
Ah.
Now into the
Okay.
following the edge.
Yes. Thank you. And then maybe if we can put points pretty often. Yeah.
Yeah. And here they're like a bed.
Yeah.
So it's
Right. Maybe a bit higher so you can see ahead as well. Perfect. And yeah, we'll just keep going across the edges. Leo, if you can add points. Oh, so we just got like a proper line perimeter going. Thanks, guys.
So I asked this one guy who was sponges because he doesn't know.
Yeah, they're definitely
How can
the
he
ones.
remove sponges to me? Yes.
They're definitely the ones, not the ones that are like, they're the ones that are like, within the like, shapes.
That's about to be an interesting shape.
This cave
And then
is
you'll have to like turn and follow it.
Okay.
Yeah, you'd have to turn left a bit.
So we simply want to be
Yeah.
turn around.
Oh, how'd you make this? Turn catch on some kind of shower.
Perfect.
Yeah, there.
And then
That's nice.
go shark and potentially a
You
cat
want to
shark.
be on this edge?
Yeah, just follow this edge here as we continue making the perimeter, and they'll keep dropping points. Perfect.
Yes. Yeah. 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 It's nuts.
All right, and then we're gonna have to turn right here. Follow that edge.
This camera can see better the turns.
Yeah.
So I'm looking down.
Can we turn right here?
Hit to turn?
Yeah. And then following this line there. Yeah. A lot of curves.
So that's the other thing I was thinking about last night.
Yeah, turn right a bit again. Following that edge. Think. Yeah. Yeah, and then we're gonna take a left essentially here.
So I would turn here
Yeah.
because it's not, that patch seems to be a separate situation.
Taking, turning left. Right, perfect. And we're just gonna go for a little bit and probably take that left again right there and follow that. Right here, come to this edge. Yeah.
Oh, when we do that.
Yeah, take that left. Yeah. Keep following the edge essentially.
Oh, when we do the transects, and then if we go close to seepage.
No, I mean like super close, but like right now we can't even see like sponges on the side.
Can
We are not looking for sponges. I'm looking for the
No,
borders
don't
now.
tell me what we're looking for or what we're not looking for.
I'm sorry.
I said we're
I'm sorry. What are you saying? I'm looking for the borders of this picture.
but we also need to be closer, Mike. Like we can't be
Can
this
we-
far
yeah,
off
a
the
little
seafloor.
bit.
I'm not telling you to be on the seafloor, I'm just saying we need to be a bit closer to the seafloor so that we can actually make out what are the different kinds of animals we're seeing at a large scale.
is about 4 meters okay? Or should we get to about
this
3?
is exactly.
Okay.
So we can see, we're seeing bubbles, we're seeing sponges, we're seeing tube worm bushes, whatever it is. Perfect.
Thank you. Alright, so this altitude is pretty okay. And then we're just still following that edge. And when we hit that top bit there, we'll take that left. Thank you.
Oh, yeah, yeah, yeah.
Perfect.
So you
Yeah.
think on this one?
then we'll have to do a bit of a right to get that little hook area. Yes, we can take that right there. Yeah, so along this edge, sorry, around that. Just so we get a gauge of like how it extends out. Crabby. So many dead shells in the distance. Yeah, so you're gonna take that.
It's standard, like, right. Yeah, like things like time, you know, like
Which is what? Perfect.
well, they're coming up.
A bit of a left again. Perfect. And then just follow that line. Can we stay close to this edge here? Yeah. Is there? I just see white on that end, so I was like, we follow this. Yeah, we're a bit. Yeah. And just again, we're following that dark edge essentially. Yeah, and just keep going around that. So it'll be a bit of a right here. Keep going, perfect. Keep following like that. Yeah, like that essentially. Maybe a bit higher for sediment. Oh, yeah, if we could stick on this edge, but perfect. And can you get back a little down if possible? I'm not sure if we're in a slope right now. And just do that.
Jeez, man. Let's get more muscles than Leo, huh?
Why?
No, not true.
Yeah, and just that edge, and then we're gonna do that, basically a hard laugh essentially, right there. Perfect. And just keep going along that.
No, I will. But it's okay. It's broken. It's mostly the test with this last. on right now, or so one's missing and the other is?
No, all do it come along.
Yeah.
As you do too many turns slowly.
So right now, is there one that has it and one that doesn't? Yes. Okay. And the mix.
If we need to be like a meter higher, potentially, to try and stop. And just, yeah.
That's prepared to roll.
Yeah. And then just keep going along this edge. I think we're getting close to where we started.
I think maybe I'll talk to the welders, we can get one molded out. I mean, that pool is super annoying. It's always in the way, it's always falling off. Should probably think of something else.
Leo, can you let me know where we are in relation to the first point we made?
We finished.
We finished?
I got you, Mike, you do.
Okay. Now that we've done that, now that we've made the perimeter, do we want to do?
Or like what do you mean?
You just run the perimeter like this. So we want to do something
Oh, yeah, just the area?
like this. So this is the perimeter of the mussel bed. We know that like the mussel bed is not the only seep, right? It's like zonation.
Yeah.
So we also want to potentially run like transect in transect setup. So a meter above seafloor, lasers on.
Yep.
Things
You might be able
zoomed,
to do.
and we run a transects, 2 transects across.
What minimum length do we want to do there?
Till we get to the end.
So we get to the end?
So
Okay,
we
no
like, well,
problem.
no, we want to start
Way out.
off and get.
So we can
off.
get these zones.
Exactly.
No problem.
And then we also, and then after we want to just sit in some random places and get zooms.
Of course. Okay. Typically when we do transects, we just put down points along the way and flashlights. In this case, I think it'll be a bit more difficult to do that. Because we don't actually.
Beer is underway. Are we finished the perimeter?
Yeah.
Okay, so I've explained the general idea to Beer, so maybe she should come in.
So look, we could do this.
I mean, we can start it, but so she knows.
We can get into position
Yeah.
for what zone you want to
Yeah.
be in.
So I think we just want to be, I would head out of the muscle bed and transition way off.
And then see how you
So-
basically see how fauna changes. as
Do you
well.
want to do like 2 like this, or do you want to do 1 like this and then like this? Like I'm looking
Sure,
at the
that's- yeah,
shapes. I'm
yeah.
thinking maybe we can go. Let me show you.
Yeah. So instead of doing this,
Mhm.
let's do this.
do
Okay.
like this.
Yeah.
And then 1 like this.
Yeah.
Okay.
Agreed. And then we, what are we doing? And then, yeah, so but in transect setup, so basically there we would wanna, we would need to monitor 100 meters and flash the lights in between. That's it. Just a flash of the lights and
I
then
would do more.
continue on.
Is it? How big is it? 1, 2,
No, so I'm
3,
saying
4,
in
5,
between,
yeah.
we run until, we run from way outside of one side to way outside of another side, but then every 100 meters we're flashing the lights.
Okay.
But normal transect setup, which is tilted down to the edge of the tree, so you just can't see the tree, which is, looks like where we are now. Zoom and lasers on.
Yeah.
And then we're not adjusting.
Got it.
And a meter off seafloor.
So I would do, let's see, so it's 1, 2, so 1, 2, 3, 4, 5. It's 50 meters across. So I think we can do,
Alright, we'll figure out how to exploit that once we're on deck. At least now it's not gonna go away like the snail trail well, right? You can also do a sonar sweep, but I don't know if it'll show these or not. They're pretty low. Okay.
it shows it in green. I mean, okay. So,
Should we get Megan involved because she's gonna lead on this?
we're making a move. Yeah, and we have to get the air involved. I mean, she's over here.
Yeah. So if anything, do we want to start positioning ourselves?
I
at
think we
the
need to
zone?
put the lines up
Okay.
so that these guys know what they're following.
Okay.
I guess-
Oh, I made a new mission, and then I went to waypoint mission, and you can just click waypoints. You can even tell it to fly along that path and stuff if you want to, but you gotta make sure you're clear because it'll run and stuff. Yeah, Breezy's is pretty good for stuff like that.
Where's the meter?
So there's one in there called, if you look, scroll, see what says main, the right, down. That, if you scroll, there should be one called like muscle stamp or something.
I know.
I know, it's not.
Or just select different ones and see which one lights it up. Maybe just send it this. I don't know. Yeah, if you have the different missions, just click them. One of them should. That one. That's the one you want to export. But we can mess with that one for you. But to make a new one, see what says waypoints up there, to the right of that. Hit that guy. It's the new mission.
Oh, this wood.
That's what else happened with the Falco waypoint. That's what happened right here. When the station keeper was still in this mission, so it connected
No,
it to
just
one.
the ground.
So it's good to have a
But we
blind
take a
mission for you to be
temperature
stationed.
measure and there was no
You don't have to.
difference.
Okay, anyway.
Okay.
Where's the boat? Oh wow,
Where's the boat?
Holy
Where
moly.
is that boat?
Freaking boat, it's getting away. Alright, what are we doing?
No, because
Okay,
we were
there's
supposed to come here, but ended up finding this on the way in, so.
So,
So we didn't even go to the good stuff yet, theoretically.
this is what the patch is, but it's not really good. It's like that. Right? So we have this
Fred,
one-
jumped in, wait,
one
what?
transect from the outside to the patch, and the other outside, and another one along the widest point, and then another one in this direction. So like this. So it would be. Sorry. So the linear transect address starts probably here and runs all the way through about there.
Okay.
And then, so you can mark it. And then another one that starts there and goes all the way through about there. So we need, this is a 10 meter, right? At least 30 meters on either side.
Can you zoom out on your map? Let's see what the topography does.
Well, it's
Uphill,
not,
downhill,
we're,
pretty flat.
well, we're not flat because we're on the double.
We on the top here.
Yeah.
Am I looking uphill or
And I
downhill?
don't know if you saw what do the drink, but essentially how
North
the
is
which
uphill?
is the most of that.
Just zoom out.
So you can see the zonation factors. When you go.
Could we do that transect then like this, that would work?
Yeah.
That would be one of
And
them.
then you
That'll
can
give you
do
better video.
That would be
If
one
that's
of them,
uphill.
yeah.
And,
Oh,
about
because
which,
you're saying that running it across is not gonna
Yeah,
work?
running across would be fine, but just if we're going this, I think we're gonna go downhill, which.
Oh, I don't care which way you go, go whichever
Start
way
here.
you want, but we want it across, and preferably not across to the edge, right?
Okay, so I'll start here first,
And I
and
think
I'll
two
go like that.
It's not 100. Maybe start, maybe it's to give it 20 meters.
This one.
So the thing is about 50, right? So you
I think
can, you know, there on both sides, so then we'll get the position, like, and then the same.
Okay.
Okay. And yeah, preferably not close to the edges because we may be seeing different things close to the edges, so we want to have our position. So, like, what I mean edges is like this as opposed to, you know, this. And then
No worries.
We got this. All right. It was lovely being with you guys this morning as we found this bed. Now I'm going to hand you over to Megan as we do some transects. Thank you, Tyler.
Here you go. Boats should be getting closer all the time.
Oh, yeah, I thought, sorry, I thought you were on.
No,
So I wasn't
no,
looking
I
at
was
you.
listening.
Okay, great, yeah. So anyway, you know how to do it.
With the flash of the light.
In the beginning, yeah, and then at the end.
Okay.
Um,
Because there's not really the public
that's
to
what I'm
monitor.
Okay.
Got
Yeah.
it.
And
We'll take a turn out as well here real quick. Little donut.
All right, good morning, everybody. My name is Megan Conkling. I'm a research scientist at Harbor Branch Oceanographic Institute at Florida Atlantic University in the United States. We have come across this massive methane seep here with a bunch of mussels, tube worms, sponges, and we just did a kind of perimeter around the entire mussel bed. So now we're going to start doing a transect through the seep area, so that way we can look at transitions between different species, habitats, and stuff like that. So bear with us while we do some science here, and hopefully we can continue to see some more cool and exciting things.
So I'll get over here, then we'll make a new mission, and we'll put the transect on it. It's 100 meter. Doodads, okay?
Perfect.
You know how to do that, Marcelo? I'll show you. And once we get over there. Share, go ahead. Yeah. Create mission, yeah, name it like muscle bed transect or something like that. Different color than we have, but not red, not yellow. It's felt like muscle bed transect or something like that.
It's transect 1. He's just hitting.
Oh, because that's
yeah.
a line.
Yeah.
You've done a few transects before, right? You know what you do?
Just
Once
my
or
first
twice,
one.
Yeah.
Yeah, okay.
He's winging it here. He's winging it.
Probably guess.
So every time I make some points,
Yeah,
marks
so
there.
now if you hit waypoints mode there, then every time you click, it'll add a waypoint. But let's see, let me just freeze here. I think the easiest way might be to go like this, right? Measure, go from us, click, 100, right?
Yeah.
So this is going to be 1/100 move then? Is that it?
Yeah, pretty much.
Oh, that's weird.
Yeah.
What the heck? Okay,
There we
The
go.
size of that arrow we prepared it.
I know.
This is not as big of an area as I thought. Okay.
You
So
want like a
100
full 100
meters
like
north
that?
would be right here, or do you want a little less?
No, we don't need it that
Full
long.
100?
Yeah,
Okay. Yeah, like
yeah. No, we'll do
like a
maybe
75.
20 meters on either
on
side
Okay.
of the boundary of the seep, so just 20 meters. Because the seep itself, like the perimeters, looks like it's about 50, 60.
So once you measure, you can right click, and then you can either add waypoint, add marker, copy position. So I just do add waypoint. Oops, I was in the wrong commission now. That's okay. But anyway. So yeah, just do that, but in the
Sounds
new
good.
mission. And that should do it. Yeah, now do you. Okay, go ahead.
So, use it once.
Yeah, there's some jobs that we do stuff like this,
Mm-hmm.
and then you have to like tell it and it'll automatically like M3 mapping and
Now,
stuff.
I'm going to
Pretty good.
waypoint one.
Just to make a line.
Supposed to be waypoints, right?
You can just right click once you measure it.
sorry.
So, yep. And then you can do another one right where we are. Boom.
Yeah, which is about where
That's it. Okie dokie. Everybody ready?
So every time you pass, you click waypoint, or just keep going?
Oh no, that's fine. Then we'll do one more in a minute that's gonna go from west to east. That'll be after this. All right, coming down. Cameras are already, should be good.
Should be good. Lasers are on. Yep. They're very tiny.
The altitude is gonna change a little just because
The slope and everything.
Does that.
Yeah.
We'll keep it as consistent as we can with the picture.
We appreciate it.
Okay, starting.
Starting flashlights.
Shouldn't even need to move.
Starting
them.
transect too. Thank you.
Did y'all do any little Charlie?
No, so the Johnny had that issue with the camera.
Oh, okay.
Some goes in, and so we took it out, so we never brought it down.
Always causing trouble.
This looks like shell and then sponges on the edge here, yeah.
But the sponge people are going nuts, really.
That's me. I'm one of the sponge people.
Sponge people.
Yeah.
It's been a sponge-tastic cruise.
It has been a sponge-tastic cruise. But we're also seeing like the zonation between the sponges and the mussels here, so.
Good Scrabble word there.
Yeah. So we're seeing a bunch of tube worms, mussels, sponges, of course. Some hydrate on some of those mussels. Looks like some snails, gastropods.
Was it like this, like the whole dive, or just suddenly kind of came across it?
I think it's just coming up to the peak of waypoint 4.
A bit slower?
Yeah,
Sure.
Have some shrimp here. Mm-hmm.
Could be one of the biggest ones we've seen, huh?
I think so, for this cruise.
It's gonna look like I'm too far away. It's because there's a rock underneath.
Mm-hmm.
You can see the edge already, huh?
Yeah.
It looks like a cliff. Such a strong delineation.
Yeah. Some crabs here. They're on.
They're on. I just said
Yeah.
kind of high because of the rocks under me.
Yeah. They're there.
Yeah.
Some tube worms more, and then we get to this very defined boundary at the edge here.
You wonder what the shell happened to.
Yeah. Yeah, he did. I give it a 10 out of 10.
It's too early.
No, it's not. It's perfect timing for that.
Yes.
So as we leave this mussel area, we're getting into more sponges here too. Yeah, it's a nice little feature we have here. Got a ghost shark swimming ahead.
There's a lot of these.
Yeah. And some more tube worms on this feature here.
I'm just about done. I think we're done here, huh? You good?
Yeah, let's just go to the edge of this and then we can turn.
Just one is sponges.
I do. They deserve their time, okay?
Okie dokie.
Perfect. And then flashlights.
Oh,
Perfect. And transect. And then we'll position for the next one.
There's a lot out to the east as well, kind of like we expected.
Further up, like to where it ends.
Oh, you want to keep going? Hold on, Mike. We're gonna continue to go out because we're still seeing like presence of seep organisms, so we want to continue.
So we just over the end.
Yeah.
Sure, I just gotta move the boat.
Yeah.
So
So we want to continue following that track and then
Start do it again, and, you know.
just coming
The thing it's easier for him to learn this, so if he's been super but you say that it's still, still stuff going, like there was still circular, it's just to add some length to it.
Okay. Even
Until
though
we run out.
Okay. Even though it's not where we started.
No, it's okay. We will
It's
just have
okay.
another one.
We'll have
I
another
will start.
one.
Yeah,
Okay.
Just for
Okay.
So if
Is it?
there's no, so if there's no, if it's dead shells, then that's fine. We can
cat
start,
shark.
but if it looks like it's still some transition, we can start it.
Yeah, it's a little
Transition,
sulfur
catch
with
a
jobs
transition.
You.
And then on the way.
Yeah.
That's
So it
terrible
should not
timing.
be 20 meters.
Yeah, you don't have to like.
Probably more.
We can confirm.
So we're gonna
Well,
Seen him before this came up. No,
Booking.
follow him a little bit if that's okay.
Yeah, if he turns towards us.
Looks like he's coming out of the muck, so that's good.
eel pout. We have a gulper eel here. We have a crab on the bottom next to a sponge. We've seen one of these on a previous dive. We're just seeing if we can get some good footage of him swimming around here. Beautiful.
So black.
Mhm.
So
black.
Yeah.
And they've got like little smidge of bioluminescence on it.
Hey, can you see if we can brighten that? Can you brighten that up a little bit, Marcelo? Bring the ISO up some, so we can see him. Hopefully he inflates his.
So you can see like the elbow of his jaw.
Can you do my focus too?
That's a
Oh, there
deeper
you go, the lateral
question.
one.
I saw one previously, with its jaw like
You're gonna have to do it manually. It's too dark for the auto to pick
but
him
this
up. I
one's
think.
just kind of swimming around.
what is it?
It's a gulper eel, so it can, it's like extend its hinge, like the of its jaw, so that way it can eat
Yeah, so
larger
that line of
organisms.
dots down his side.
Yeah. I know.
He's cool, but he's not really.
No, you're good.
Yeah.
That's
It's jaw open. It might be like a
He
bit more
didn't,
unusual.
he didn't
Yeah.
pop his jaw out.
He didn't want to pop out. Could you ask him to?
Yeah, I mean, sometimes they
Trying.
just do. Yeah.
Trying
I'm trying to tell him he doesn't want to listen.
Yeah.
Maybe drop some food for him. You can open.
Yeah.
Give him a chocolate or something.
Musketeer.
Wow, look at the
That's
detail.
kind of cool though.
Yeah.
It's fun swimming with him.
Beautiful.
It's called a golfer.
Gulper. Hard right. That was tiny.
Yeah, he's a itty
Yeah, itty bitty one.
Itty bitty.
Yeah, we can run up to your side if he's just gonna keep swimming.
Okay.
Thank you. Some fun shots of going up the muscle side.
Later, dude.
Beautiful.
Beat it, hit beat it. Look at that crab. Jeez.
Yeah, it's a good
King of
size
the
one.
mussel there.
Yeah. King of the mussels.
All right. Anyway, back to work.
All right, yeah.
All right. So we're gonna do the same transect, but we're gonna pick up on the north side and keep going. Is that?
Yeah, we want to try to find the edge. So we're gonna kind of try to get out to an area there where there's no tube worms or associated like organisms with seepage. So
Sure. Not even gentle seepage, right?
no.
Oh, it's real rough. That's a rough one. If I don't feel like it's
so much good stuff.
yeah, if I don't get a specific request, it just is what it is. You're getting one.
I've got like 3 deep sea bucket list items. Like this last week alone. That beach, Savannah, I know. I know. And those troncos. Oh yeah.
I'm sad I missed that guy.
I know.
Those are cool, man. Hey, you want to move the boat about, I don't know, how far you guys want to go after this? You think 20 meters, 40 meters?
Probably let's start with 40 and see. Hopefully.
You move the boat 40 meters north. You can just tell them that too, I think, to be able to do that.
Yeah, because I think we can like go and see where we don't have anything.
Yeah.
Bridge, ROV, can
Beside
you move
there.
the vessel 40 meters north?
You let me know. 'Cause
Can we see like only?
no
only
sponge, just sediment. I know. I'm just like. You just shout
Yeah,
out.
I think it can be like dead shells, but
That
see,
shelf?
like no tube
That's where we
worms.
were.
No, we
Past
finish
it?
like around here and then see that there's still like
There's still sponges in
some
the
tube worms, sponges.
Yeah.
So we have to go even farther.
Even
away.
further. Okay.
All right, here's where we were.
Yeah.
So I just got to wait for the boat now.
Okay. Oh, he's got these yellow cryptic sponges
Why are they called cryptic?
because they're so tiny, but they actually have a lot of like environmental impact because there are so many of them. So they're very important to ecosystems usually, even though people just think that because they're so tiny, they don't have an impact. So we've seen those in a couple other sites, seep sites. Some beautiful tube worms out with sponges. Two different types of tube worms here. Beautiful.
Certainly leave their mark.
Yeah. Beautiful.
Okay, I think they're far enough here, so.
I think we have to go even further away. Or are we starting?
No, I think he's just readjusting a bit.
Okay.
We're waiting on the boat.
Ah, okay.
Yeah. It's too early. So we're gonna continue on, and then we'll start a new transect coming from the north to the south. So.
Both ways, okay.
Yeah.
I don't think north to south would be as good of a view.
A view?
Because we're facing downhill that way.
Okay.
Give it a go.
Hmm.
Okay. So you want to start
Yeah, so
from
when he
south
stops,
to
just
north?
like, pretty much. Because that's where we stopped. And then just
Okay.
continue this,
Okay.
and then have that be like the 100-meter like,
Okay.
transition.
For her just
Yeah.
to mark another end.
Yeah.
Yeah.
Yeah. So this will be one. This
Makes
will be the
sense.
third one.
Yeah.
Okay.
And we make a note.
Yeah, just make a note of the start of it now. Okay. So then we'll just use this, and then we'll start the west to east one when we're done here. So we're seeing some stony corals in the rubble of tube worms.
See you, buddy.
Because the beginning is the end of the we just like extended a bit. So when the flash lights, we just count behind and
This is
Isn't that good?
25 meters past.
A bit more. Okay. A little bit more. That's good. Okay. All right. Flash your lights, and that will be the end of that. All right. All right, and then we'll position for the west. Okay. And we'll just follow this. Kind of keep the same terrain, hopefully.
So now I want to go like here to here.
Yeah.
Alright.
So,
You
but
can,
it-
move the boat back kind of where they were then. And I'll start going south.
So right now we're exploring a methane seep at the top of this ridge here, and we did kind of like a outside boundary with the ROV to survey the area of it, and now we're doing some transects through the methane seep to look at transition zones. And we're finding lots of sponges, tube worms, mussels, stony corals on the outer edge, as well as crabs and shrimp. Lots of different rock features here.
Yeah, go ahead. Just move them basically kind of back where they were. They're going to be too far north for this. Now I got to go like this. So if they're like here, it'd be good. A little more that way because I'm going right around where that green. Yeah, right around there. Perfect. Bridge, new waypoint. Start this before there's any shells and stuff?
Yeah. If we can just kind of go as far out as we can. Yeah. Hopefully not. There's some large rocks here. Oh, beautiful coral here. Yeah, this is good. The starting point. From Britain south and then start.
Just over this ridge.
Yeah.
Another one.
Cat shark.
Do you
Look
think
at
this
that
is
color.
like
Wow.
if he just swings it around?
Hold up a second.
Is that good? Even
Yeah,
there's
we
still.
don't have two drums.
Yeah. Okay. It's all pretty dead.
You got subsea slot canyon.
Yeah. Look at that.
Oh,
Beautiful.
corals.
All the corals taking advantage.
Yeah.
Perfect.
Alright, so facing. You can. I'm just gonna truck till they tell me to stop. But it'd be good to shut the lights on and off, I guess. So yeah, do 100 meters for me.
We are going to flush. So
I do not know what is gonna happen with the terrain during this, so there'll probably be a lot of.
Yeah. Oh, for sure. Yeah, I think it's just on the outside boundary on the other side.
So yeah, you're still in the muscle bed one, so just measure from me. 100 meters to east. Yeah, oh perfect. And you're right around there, just right click. You should be able to add waypoint. You're on the line, so you just gotta go not on the line. It's gonna be hard because there's a lot of lines there.
If we want to move it. Yeah, just do it.
If you zoom in over there.
yeah, because I mean
You can, or you can right click.
it's blind, so it means
Yeah.
I mean in this case specific.
That waypoint. Honk, and then add another one right in that where that was.
Okay.
Yeah, right there. A little farther.
What kind of
A little
coral
farther.
did you think that was?
Source of black
Black coral.
Trouble. So 100 meters was like right here, right? There you go. Find that mission you're in.
Muscle bed extent, I think.
No, that's the
No.
that's the outer.
That's the green one.
Oh, okay.
Put the green one.
Green one.
Right?
This?
The green. Right, green one.
No, that's see, it's got so many. Shouldn't have that many. I think you only have like 3. I'll just do a new one. So you want me to. So. Just gonna do it here. Okay.
Perfect.
Everybody good?
Yep. Flash your lights and then we'll start.
Alright.
Perfect. So we've got a black coral, some stony corals on the edge here, as well as squat lobster, crab.
Falling off the cliff here.
Yep. So we try to go to the outside edge of these kind of ecosystems, so that way we can do this transect to look at different zonations along the seep area, because we've seen some very defined boundaries being formed between mussels and sponges, but there might be other kind of zonations occurring.
Another gopher.
Hmm?
I just said it's slower.
It's slower?
Yeah.
0.1. Okay.
So then when you take the frames,
Mm.
the little things just like move lower.
Yeah, okay. We're starting to see more sponges here. Is there any direction for after the transect?
Yeah. Want to see from different spots. Around.
The bubble spots?
I don't really see all the bubbles.
I mean, just to see.
Yeah, it's random spots.
Yeah, we might collect some muscles for anything. Elephants.
We have some live mussels, tube worms. So once we finish these transects, we'll actually come back to the seep and at a few spots sit down and actually get some zoomed in shots to see what is going on at the finer scale at the seep, and maybe make some collections.
Where are all those waypoints to the east there? That's like 572 or like zigzagging around. They're like bubble spots or something.
Yeah.
Oh, look at all the shrimp right there. Oh, man. Shrimp city.
Looks like we've got some hydrate on the mussels here.
It's hydrate to what?
Mm-hmm. Yeah. Nice little dusting. Which usually means it's a very active area right there. Got some squat lobsters. Yeah, it's a massive seep here.
Keep going past this one too.
Yep, correct. Tube worms. Starting to see some sponges. And this is where we're starting to see that zonation between the mussels and sponges here, and then sponges throughout this expanse here. And lots of tube worms.
Just like that.
Yeah. And we got some stony coral now. We're going to continue going here. We're still seeing presence of tube worms. Oh. Okay.
Yeah, let's keep going a bit.
Yeah, we'll continue on a little bit further.
So we're seeing worms.
Yeah. Try to get to some soft sediment. We'll try to get to some soft sediment again.
So what was it, 100?
That was just 100 mark, yeah. Lots of tube worms here. Can you slow down a little bit?
Yeah. Rocks coming out.
Okay.
come back after this, right?
Yeah.
I'm gonna try and not use it.
I just feel like this is the edge of a mound.
It certainly has some interesting.
Yeah, that's what I'm thinking. So, yeah, we'll end it here.
Some interesting topography after this for sure.
So, like, a little. Continue going a little bit until like the edge there.
Something white out there.
Yeah, we'll just take a peek over this corner here. Got some serious rock structures going on here.
Something to the right, I think, but I'm not going to turn because I'm transecting.
Mm-hmm.
Looks like another muscle bed over there.
Yeah. All right, we'll end here. So flash the lights, and we'll end transect and return. End transect.
Okay. Hey, can you slide the boat like 20, 40 meters to the east? You want to go look this thing out, right?
Read you the waypoint.
Oh, we have a black coral with spot lobsters. Mm-hmm. Crazy. Oh, hello. What?
We're not transect?
No, we just ended it. So we're headed back to the sea. So.
Look around the fishes.
You want me to stop if I see one of those again?
Sponge people.
Yeah.
Yes, please.
Transect. Oh
Back,
my gosh.
From the back.
Giuliano, yeah,
I will
backseat
take
driver.
control, like,
Yeah, I know.
full chain.
Just give it 10 minutes, bud.
Alright, you can pause
You
and
want
move.
4 spots? 4?
Get back.
Read RV, can you pause the move?
I would say,
Like one on the south end, one in the middle, and then on the east end. Okay. So we'll hit the east, or whatever's easiest for you. Ooh, lots of hydrate. Tons of tube worms, sponges. Wow.
They're all sponges, the little.
The like gray stuff that's, yeah, in between the tube worms, yes. Got some crab.
Big crab?
Yeah, big crab.
Alright, turning towards the patch here.
Okay. And we're gonna sit down at 3 spots. One in like the east area, one in the center, and then one in the south.
Sure.
Whatever's easiest for you to get to first.
More patchy little muscles.
Mm-hmm.
Is that another big bed? It is.
Yep. More muscles. A different bed.
That's a different one.
Yeah.
You want to look at this first, or?
Wow.
You wanna look at this first before you back, or? We'll just do a flyover, and then we can come up to the south part.
This guy's huge.
Yeah. Just keeps going and going.
Huge.
Yeah. And we do have
Mussel
some.
beds are this big.
We have some bubble spots just south of this, so that's probably fueling it.
Well, I don't know where it was. This might be bigger than the other one.
Probably.
I mean,
Just keeps
you
going and
can.
going.
How big
Yeah.
will it be, Mark? I think we're gonna have to fly the extent of this one again. Look over
Yeah.
here.
Just over the edge here. Yeah. Wow. And the different colors of shells is indicative of like the different generations. So the lighter the shells, the younger the mussels are. So you can see that like this is probably the general like beginning seepage area because we got some hydrate, some younger shells, and then on the outer extent we have the darker mussels, which are usually the older ones. Crazy. Tube worms.
Yucky.
Yeah.
What now?
We're gonna go back to the main one and do 3 different sit downs to get some zoomed in shots of like what's going on at on the mussels and some potential collections.
Oh, no.
Small little bed here, and then we get back into the main area. So maybe we'll stop like over here. Maybe like in this area.
Spin around for you.
Perfect.
Face those little ridges. Now that's that spot. I think you're about to point out.
Yeah, right there. That works.
Park here.
Seems like a good one, yep.
Gently park here.
Get some good zoomed in shots of what's going on here.
Are we gonna collect here or just sit?
Sit and possible sections.
I'll start with sitting and we'll figure it out.
Looks like we've got sponges, shrimps, mussels, some tube worms.
I got you.
Perfect. Squat lobster.
What do you want to see? We got all that stuff in front of us, and then we got two worms back there.
Yeah.
A lot of cool stuff. Tons of shrimp.
Maybe we'll do like a zoom in here.
Neighborhood.
Yeah. Check out the shrimps. Got some gastropods.
You can collect some mussels here.
Okay. How many? At least 6.
I'll say 3 scoops.
3 scoops. Okay. We're gonna try and collect 3 scoops of mussels here.
3 scoops of pumpkin.
Yeah.
And those are gonna go in a bio box, are they?
Yes.
Yeah. Maybe one of these.
Yeah.
Oh,
Okay.
So what I want to
All right,
try
so I got a
is
porchetta.
like
There's an
clear
easy.
an area of mussels
Okay.
with the lasers and then have an estimation based on that how many can
Oh.
use them for the dinner. So
Okay.
if the scoops can be from the same place, or
Same
it's like
chunk.
in the same area,
Okay.
you can also
Do you think like that central area here? That's gonna be possible here?
Yeah.
Even though there's already open area?
well,
It's okay that there's open area there? Or?
it's an estimation.
Yeah, okay. So we're gonna try to do 3 scoops from the same area if possible. And then we'll take a picture after the scoops and collections have been done to try to, with the lasers on, to try to get an idea of the area that those scoops made up. That would be great. Okay.
Pitch in and elbow up. You pitch towards us. Okay. And just elbow up. regularly, or?
Get them all from the same area would be great.
By the rope, it ought to be able to set it flat, you know?
That's good.
Okay.
Maybe it can be easier from here. I don't know.
No.
But we can always do this in different spots.
Okay.
Find answers.
If we can, if this is too difficult, we can do this in another spot. But if we could try to get a scoop, like,
Because when they are vertical, sometimes it's easier.
it's easier to get them in, yeah.
Okay. So like a scoop maybe if we can from that area right behind.
Yeah, if that's like a better angle.
Just above you. Kind of in there, huh?
Yeah. Perfect.
Sometimes if you give it a little jiggle as you do, it'll get in there a little better.
Perfect. Wonderful.
Now,
Your preference is to where to go?
Sorry?
Preference is to where to go?
Yeah, I think it's easier.
Okay,
to
bio
use.
box 2, A or B, whichever is
Two?
easiest. Yeah. What's the sample number? 448.
4,888.
Thank you. Perfect. We can put the scoop away. Oh no.
Put the scoop away.
Yeah. That'll be good for now. Okay. I'm gonna hand off to Juliana now.
Morning, guys. Good morning, everyone. Buenos dias. Bom dia. Bom dia. My name is Juliana, and I'll be with you for the next hour. We will stop for fish now. Hola a todos. Ya. Los que nos escuchan de países hispanohablantes, buenos dias. Também o pessoal do Brasil, se tiver, bom dia. Yeah, there's a lot of everything around here. Are we now bringing some of those shrimps?
So?
Are we bringing some of the shrimps now?
What shrimp are we doing?
¿Queremos los camarones también?
No,
Solo los mussels?
we just need to put when they finish to put the scoop some lasers on the place that we took the mussels.
Ya.
from.
¿Y luego nos vamos?
Ajá. Y lo que estamos haciendo es
No me
que
dijo.
ahorita estamos
¿En este lado?
no ahorita estamos ahí como en ese patch. Queremos sentarnos en al menos 3 sitios.
3 sitios dentro de ese patch.
Ajá. Esto tiene forma como de una bota inversa.
Ya.
Que vamos a estar en los pies, en el talón
Y en el...
y luego nos movemos.
I like it that way. Yeah, I like it.
Sale y vale.
Just gives you a little more angles you can do, you know? Kind of like that, still work that. Yeah. I mean, just see what's better for you. You don't have to. Just works well. It's different working so close to the arm. You know, you don't usually do that in other industries. Okay, moving on.
Yeah, can we just get a zoom with the lasers on where we took the mussels, and then we can move on to the hooked dough. Stay there for like 10, 20 seconds. Yeah, that's great. Yeah, let's just like stay there for a bit. Fight.
Let's film those shrimps to have a video of the behavior.
They are.
Yeah, let's just like film this for a bit. No, it's. I think
Yes,
it's.
yes.
Yes. Okay.
Okay.
There's baby shrimp as well.
And amphipods.
Is that baby?
No, those are amphipods.
Ah, sorry. There's no baby shrimps. They're amphipods. I got excited.
I guess they're not gonna fight.
They felt observed.
One job. Eating though. Okay.
All right. Wait.
Did we get the card?
Yeah, that's what it is.
The what?
Oh yeah.
What kind?
Thank you,
Zoom
Alex.
on in.
Here.
Middle.
Here.
Yeah.
Oh.
Right. The middle. You have
We can grab him.
That's a nice one.
You wanna grab him?
Yes, please.
All right. Cucaracha de mar.
Alright, that one might be a little tricky. So you're gonna get the arm with the soft jaws. And you're gonna have to kind of get your fingers in there and grab that shell.
So we saw the chiton,
I can move closer.
chiton in español, and at least in Spanish we call him sea cockroach, cucaracha de mar.
Just so they can get on.
We actually have seen a couple but not collected one, so we're gonna do the mission, which might not be as easy as it looks.
Yeah, right under all those things. So you might have it coming from the top. A real shiny one there. Try not to smoosh the chitin if you can help it. All you gotta do.
Para aquellos que van a Acapulco frecuentemente, en las rocas se ven muy seguido los de superficie, negros, adheridos a la roca y son dificilísimos de quitar. Cuando estaba haciendo mi licenciatura en la UAM Xochimilco, los estudié durante un tiempo y me pasaba horas intentando quitar uno de la roca.
It's like you got the reach probably.
Yeah. I can volunteer to love.
They also challenge you more so.
I can volunteer to love. This place is epic. Before you even ask. Oh. We are discussing whether we extend the dive here.
This guy gonna go in the same bio box? Or somewhere else?
We can put. We can, no? Yeah. Or mussels and a chiton. The crab in the back is having a traumatic experience. There you go. Yeah.
Spotty in there.
Yeah, it's having like a. Huh? Oh, I could only see the. Sorry, everyone, again.
That's okay, they just want the chitin. As long as he don't fall. Shell is just as ride. Alright, you said by same bio box? Different?
Yes, please.
Which not really?
BB2A.
2A, got it.
Not fierce. They're not definitely not lobsters.
Can you lift your arm up a little bit? Alrighty. Open 2.
¿Le diste otro número?
Cuatro, ocho, nueve.
Sound good. Thank you.
Gotta remember to cut that one little green rope. Okay. Ready to go?
Yes. Anything else in here? Beatriz? Anything else? Can we go? To the al talón? I think we can go, Zach. Thanks. To the heel. Oh, sea star.
All right, now we're going to the central area.
Yeah, to the like, if this is the boot, yeah, either the elbow or the heel or whatever shape we decided it is.
Make sure there's macaroni in it. That's one of those garden hoses.
Yeah, okay, I can see that. Yeah, but they say it was a boot. I'm like, I don't know.
The upside down boot.
Well,
Look at all the shrimp.
yeah. Yeah, notice the difference of the amount of life that we perceive when we're just doing a flyover versus when we stopped. If we like stop here, we would see so much of the small details moving around. So that's one of my favorite things, is just stop and focus the camera in a small place and just look at that. Because you like, with time, you see your eyes kind of identify and get used to it, and then you start seeing so much different life in that
Drop off.
small space. Yeah, big drop off.
Well, we haven't seen bubbles yet, but that's okay. Like bubbles are just like extreme seepage. Like you can have all of this extensive stuff, but we've never seen bubbles ever. Yeah, it just like seeps out like a fluid.
I think this is good.
We like that gentle seepage, right? Alright, so somewhere in this neighborhood?
Yeah, I think we can like look around here.
Weird patch right below me. See it in the sit?
Oh yeah, the white.
That stuff.
Why don't you just say like
Shoot at that real quick.
Is it like clams or is it bacteria mass?
good job?
It's a little dead spot, I guess.
Oh yeah.
Weird. Alright. So go right in this lighter colored stuff here. That is good for you?
Yeah. Anywhere here is good.
Yeah.
Very marked age structure in here.
Very what?
Marked age structure. These are juveniles. You can see by the size and the color versus these ones.
So would that indicate like the seeps moving or something?
Maybe like the expansion of the population towards one side.
Después de este, cuando nos vamos al tobillo, también si vemos un sitio que tenga gusanos, o sea,
Ya.
que no sea una cama de
Ya.
hormigas.
Un par. Oye, ¿quieres juveniles o adultos? Mussels.
No,
Ah, no, solo vamos a observar.
sí, vamos a observar.
Yeah.
Sí, vamos a ver qué tenemos.
So can we start by zooming in this patch of like juvenile ones? Stay there for a second. Look at that. All the juveniles. Do you think we can get the lasers to be in the same shot? I want to see how small this. Yeah, look at that. Remembering there's 10 centimeters between the two green points. So these are really small baby mussels. Squad lobster, some shrimps. I mean, let's actually stay there for another 10, 15 seconds. An eel sprout on your right. So this is what I'm saying. You just like stay in one place, and then things just. I feel like things are getting busier and busier and busier by the second. Another eel sprout on the down left. There's a lot of going on. Shrimps, polygons, and this is probably like a not even a square, not even a meter by meter space. This is like a smaller space. We see so many life. Okay, we can move to the next one like that. Wherever that. So the right side of this image, the lighter mussels are juvenile, are younger mussel, whereas the left side, oh, look at here. Let's check it out here. The left side is morados. Yeah, let's just stay here for 20 seconds. Yeah, that's perfect. All right, how many string people see? Okay, I counted at least 40. More than two. Say that. More than two, less than 1,000. Somewhere in between. All right, somebody said 32. Good eye. Thanks, Zach. Yeah, so. Oh, there's a cave. Who lives there? Yeah, they seem to really like that. Whoa, there's even more in here. Shrimp condo. Eel sprout as well. On your left. Bottom left.
Big time?
Big time.
Alright, nothing to report.
A couple of tube worms.
Have the roasted veggies.
Also on the left.
Is that the hot tip for the moment?
So we decided that this mussel bed is a garden. Got this praise. Yeah.
Morning, Sunny.
Morning.
E aí, Mike? Tudo certo, você?
So what are we on there?
We're just looking at shrimp.
Alright. Enjoying the view.
Enjoying the view. So we're currently, where's my hat? So we're currently kind of like going back to these gardens, right? That's it. So we're gonna head down. We're not collecting anything unless we find it interesting. We're just like doing a visual inspection. So after this one, we can head down to this somewhere in this area, land again, and then just do a visual
Okay.
inspection. And I think we're good to move.
Good to move.
Whenever you're ready, yeah. ¿Quieres ver algún otro pedazo de acá? Yeah. We can zoom out as well while we move. Thanks. Sonny. Yeah, that's good. That's good. That's good.
Good.
É, estamos indo. A gente já fez tudo isso ao redor. E aí a gente tá vindo pra cá.
Esse ponto quadrado.
É, não, essa parte aqui de baixo.
Só pro meu.
É. E aí a gente vai pousar aí pra ficar um tempinho, cara.
What are you, General View?
Good morning, Mike.
Good morning, Pete. How are you?
Fantastic. Yourself?
Great, thank you. New waypoint, please.
I see. It's okay. I'll head there now. Thank you.
Thank you.
Lots of dead ones in here. Interesting. Right, so then if we can like turn kind of like left or even around our edge and then fish. And then we can land somewhere around here. That's perfect, Sonny. Thanks.
You want to line where the dead ones are already?
Yeah, it could actually be, because then we can see some of the dead ones too. Well, there's a, I already seen a big crab. And if we see a worm, we'll take him ideally. Thanks, Sonny. So let's do some observations. And we start with the big crab right in front of us. Look at that guy.
The delays on there.
Yeah, they're
Top on,
yeah, they're here. Bit bigger than 10 centimeters crab. Yeah, let's stay on him for a bit, please. This is one big crab. We haven't, at least, well, I've been here only for an hour and I have not seen this crab yet. Oh, it's in the move. Let's see what he does. Look at the movement. He is able to actually walk on top of the mussel bed, which must be so really sharp. Crab on a mission. Sí. Oh, he's coming closer to us. All right, thanks, Sunny. Can we? There was a point where some, I think, yeah, it's getting those eel sprouts that I saw. I see them now, but can you just let's pick up like a spot to zoom in and see what's up. Just pick one. Oh, here. Found what I wanted to look at. Yeah, we can just stay there for a second. Thanks, son. All right, so we get some eel sprout, which is actually a fish. Very cute fish. Look, this mussel is actually, it almost looks like it's opening and closing. There's also tube worm on the right. Not a lot of shrimps in the side of the mussel bed, interestingly. Especially compared to the other one.
This guy here.
Focus.
Yeah, the one with the large legs.
This guy here.
Yes.
Can we try to focus on that one, please?
It was full zoom.
Yeah, just the focus. Oh, he's on the move. Okay, it's Beatriz. ¿Sabes? Wow, that is some. You guys can experience the power of this zoom. This animal is probably like 2 centimeters size, 3 maybe, probably less, and like look at how defined we are looking at it. The amphipods are like, yeah, the amphipods are 5 millimeters. Yeah, so this is like,
Okay, well, thank you.
yeah.
Did you see the, was it Chernocop's today? Fish? Maybe film?
Oh, yeah.
No, I did not. I missed that.
Yeah. What do you call it?
It looks
Toad?
like a little frog.
Yeah, like
Yeah, I
sea
know.
toad.
I missed it.
Yeah.
Oh, what?
Yeah, he runs on the seabed with his little
Paws.
fins.
Yeah.
But he also swims.
Wow.
Yeah,
That is amazing.
okay.
Okay. Do we want to see anything else in this patch? Can we zoom out a bit, please? Or we're good. ¿Quieres intentar este? Can we zoom in here just to see if it is worth moving?
We could reposition.
Yeah, we can reposition if you want.
What is it, the blue one?
Is it worth? Yeah, can you zoom in here, Sunny, so we can try to figure out what it is from here. Looks like a tube worm.
Yeah, it's a good one. I think we will see them in better spots.
Yeah, so it's good. Okay, I just thought you wanted one from exactly here. Okay. Are you ready to leave this
Yes.
amazing place? Are you sure?
It will be the same all over these
Okay.
waypoints.
So,
So I think we can like go through
where's my...
this edge here. These waypoints.
Yeah.
And try to see if there's a spot with big tube ones where we can also sit and make some close-ups. And then we can start going north to waypoint 5, 7.
So you want me to come here first?
Yes, please.
Okay.
But you can fly like maybe east,
Yeah.
find the edge, and then we'll start going north.
Sounds good.
Thanks, Sunny. When was the toadfish? Like at the beginning?
Yeah.
So if you're joining us, there's a very cool fish that we saw earlier in the dive.
Oh.
Kind of like at the beginning. It's toadfish. So worth going back and looking for it. It's a really cool fish. Eels, prawns, shrimps, mussels. Bucks. Back to all the shrimp land.
Would it be safe to eat these mussels?
I don't think so. I do not think so. They also, when we process them in here, they also smell pretty bad. Yeah. Oh,
Stop these worms there.
Yeah,
there we go.
that's
Yes,
a really
please.
nice spot.
Interesting that it's one patch where we find the two worms. All right, we're landing to see some tube worms action.
No, no. So,
Thank you. And there's our on. Eel sprouts. So can we go closer in here? Just because we can, not because we need to go closer. Yeah. No, that's perfect. Thanks, Mike. Yeah, that's great. Thanks. Right, so this is the actual tube worm. You can see the pinkish, reddish pinkish organism coming out of the tube, and they would actually be buried and will be like super long. What we are seeing is just the external part of it, but they will be really long buried in the back. Some of them are empty, some of them are dead, and some of them are alive.
I mean, just not zoom in, but focus on that.
En la parte de abajo. Can we change just the focus?
you could zoom on until if you want.
No, it's okay like that.
Oh, that would be great.
So, I'm gonna give him the opportunity to
Zoom in. No, so in here.
It's closer. So
It's closer, yeah.
So if you pull down, so the tilt, yes.
Okay.
that, and
Thank you,
then
I'll
left
do
and
that
right,
for
and
sure.
then zoom in, zoom out,
Okay.
and you let me know when you want to focus, and
And-
I'll
okay.
focus it.
Thanks. All right. So where were we?
So for you, zoom out, find yourself, then you start search for you.
Look at that guy.
Zoom out again, please.
This snail, yeah. There's actually a bunch of snails. ¿Más, Beatriz?
Sí, no, tal vez en los gusanos nos quedamos unos 10 segundos solo para poner un buen video de los gusanos.
En este pache tal vez, mira.
Sí, ese que está ahí, tirado.
Oh. If you notice, one of them retracted with the shrimp movement, the ones on the right. Eso es algo. Eso es alguien. ¿Dónde está? Wait. Don't move. 1 second. Abajo de este. Este brother ahí. Yeah. This guy. Oh yeah, look at that. Beautiful. I don't know what it is, but it's something.
So 10. 10.
Okay, thank you. Do you want some more tube worms?
No, I think we're fine for now.
Great. So then, can we zoom out again just to see if there's anything else that we want to focus on?
We can maybe reposition to a different place, just like.
In this part?
No.
Or just head up north?
In a different place, yeah.
So you wanted to go here, right?
Yeah.
Okay.
Yeah. Thanks.
Yeah, maybe we can move to waypoint 5.
After the...
And if we see a spot that we want to sit, we will tell you.
Right.
Yeah.
Interestingly, an interesting flat area. Oh, look at that guy on the right. Is this a?
That is a sponge.
Is it a sponge?
Yes. Yeah, we have,
Jeremy?
we have collected the sponge.
And a crab. Do you want to land?
Wait. Just a sec. Not sure if we're gonna land here. I just put a new waypoint.
No,
but
No?
Let's keep going.
Okay.
In a different
We'll
place.
keep going, Mike. Thanks. Sorry about that.
Sorry, Bridget. You can start to move to the new waypoint. Thank you.
There's many of them. 2 crabs. There's 1 crab here, another crab on top. Many sponges. Oh, they're retracting too.
the swoop. And other type of tube worms, all retracting with the ROV approaching.
Yeah, actually, we need to collect some of these.
What?
Of the tube
Some of
worms?
these tube worms, yeah.
We might as well just do it here.
If you
There's
see a place that you find something that you can grab easily, we will.
of the tube
make
worms.
that.
I mean, we can probably grab this whole piece here,
Okay.
because they're easier to grab when you grab like the rock. What do you guys think? Yeah.
What are you, Jerovid? Please, may you pause the move?
Okay.
Thanks, Cap.
Thanks, Bridge.
It's like we collected some, but they were dead.
There's a whole logistic between the ROV and the bridge of the Falcor. They need to be in constant communication of the movement. And so when we scientists are so excited that we're like, let's move. Oh no, wait. No, move. No, wait. It becomes a bit challenging for the logistics of the navigation. Fortunately, both the pilots and the bridge are very, patient. Yeah, see, because there's like some of them that are alive, but some of them are dead. And so it's easier if we take the whole.
Do you want to try with the scoop or the jaw just as a...
I would try with the arm and just pick up the
Yeah. So like, almost like a clamp from, sim, e pegar todo o pedaço.
We just need to make sure we get live ones.
Yeah, so this one is live, this one is live. So if we go on the
but
side.
the patch next to it has more live ones.
Okay,
That little, yeah. So I don't know if we can,
this one is this one?
if we can just try to break off, or that one up there, just try to break off a big chunk of it.
You mean the right side?
Correct.
So my
So
experience
it's either left
this
or right.
one, because there's more live individuals in here. Or
I'm
the,
gonna need a poke to see if it comes out together, because we cannot see if it's one rock or.
yeah, okay. Because if it breaks out, that would be
Yeah.
a good one.
Okay. Start on that one.
Yeah.
Thanks.
What's the most important part for you guys?
These guys here, like the worms. So see,
Tubes.
the tubes. So like you see how they all there have
Feather.
this feathers. So that's what we're looking for.
Because I'm afraid to come from this side and then get lower on the floor and then smash the other guys here to have the angle. I'm gonna try just lodge first. You think this is a rock?
I don't know. They might be just see all of those that came out in.
Even if it's not a rock and it's attached to the seafloor,
It will
it is
like-
a concreted mass. So
So
like
it
you
will-
can see.
Yeah. So it will come as like a one piece. Yeah.
But it may need
There you
to
go.
be right.
There you go. So if you
if the back there loses, it's fine.
To break it out.
I think once you get that part,
Close it.
that's perfect. Just close it and there you go.
Okay.
Yeah, got it. Perfect. And so we can put it in.
Straight to the box.
Yeah.
I'm not happy.
We're gonna try a picture, a zoom in picture for Sydney, please. And then we'll put it in. Do we think that will fit in one of the
Yeah.
BB3? Oh,
Let's take the glamour shot on top of the just in case it falls. I'll open the lid there with
oh yeah, thanks. Good shot, sorry.
So you just position it just over it and then we can take the glamour shots.
Falls in the far side.
Perfect. Yeah, good shot. Thank you.
Decrease the ISO.
Oh, all the tube worms. Is it a focus? Yeah. That's good. That's good. That's good. That's good. Thank you. You good? Yeah. Thank you. All right. So.
Sonia, you good if I reset the audience?
Yeah, I don't even use that.
Yeah.
BB3A is good. Some pieces already fell in there.
Yeah, it's good.
Valeu, Mike. All right, good job, guys. Thank you. Warm plus 1 second sponge. That went to BB3A. What's the number? 4. 490. All right. Well, I think we're getting collection here. Thank you. Okay, so we can continue to towards the north. Please. look at that sea star over there. White sea star. Lots of tube worms. Oh, no, just they retract the tube worms. It's just. Fun. Wow, look at this sea star. Beautiful. Oh, and it has. Oh no. No, I don't think we want to collect sea stars. No. Just 1 second, please. It has. It almost looks like it's has holes for air. Wow. No, not really. No, this. No, this whole like last one.
Yes. How is that image looking? Is it too white or
This is the monitor view back now. It still looks alright up there.
Are we in position to collect them?
Do we need to move to? No, I think we're in a position to collect it if we want.
Yeah.
Yeah.
Until BS says otherwise.
Are we in position to collect this one, Mike?
I might need to move a little forward.
Oh.
If we,
So yeah, it will require a bit of a movement forward. We're still looking at it. And there's another one too, no, right next to it. Yeah.
Okay, she says BS says collect.
Collect it. Okay. So there's two, whichever you guys think it's an easier collection.
either one, your choice. The bigger one or the smaller one?
The bigger one, then, please.
Yes, let's move. Let me move forward a little bit. I think you're a little too far. Let me move forward. So let me jump in.
You prefer the one further away from there?
No, the bigger one. The one
The
here.
bigger one.
And closer to us.
Yes, please, Mike.
Soft drop.
I think the soft grab can also work here. You can put it in any of the BioBox BB 3B, C or D. Well, should be small enough to fit in one of those. Such a beautiful day.
Yeah, that
Yes, that one, the first one, Mike, please.
Delicate is the center.
The, the center of this, yeah, the sea star is. I mean, I wouldn't say all of them are delicate. This is slightly more hard than a sponge, for example. But it would not like break on you. If that's what you worry about.
What about it?
Yeah, that's good. Pronto. É só fechar que ela vai vir. She's just resting. She's not attached to the. Sea stars have actually an amazing property of regeneration. So when you see the sea stars very well defined, whether like 5 arms long, sometimes they will drop the whole arm under stress and regenerate. And this is a mechanism for predators. So if a predator comes and wants to eat it, they will drop the arm, and then the predator will then focus on eating the arm, and the rest of the body will leave, and then they will just regenerate it. And they can do that even with multiple arms eaten and from a small central disc. So they have a central disc, and as long as that central disc has enough cells, they can regenerate eventually.
Okay, burrito ready.
Yes, perfect. That's good. Thank you, Mike. Sydney, let me know when you're happy with the shots. That's good. That's good. Thanks. Let me know when you're happy. Huh? Can we get up a little bit? Like a zoom out, so we get the whole animal. A bit of zoom out. Like a nothing. There we go. Perfect. Thank you. That's good. That's good. Thank you. Good? All right. Thank you, guys. So you can go. Are you doing BB3B?
Yeah.
Perfect. So collection of specimens is very important from a scientific point of view. We, unfortunately, some specimens we actually need the organism to study multiple things, from identification. Many of the collections that we're doing are first to science, to other things like pollutants, formats. What number are we at? 491, etc. So that's why we collect them. My time is up. I will leave you with Shirley, aka Sponge Master, and
I had my day yesterday.
she had her day yesterday. That is true.
My day yesterday.
And I might see you later today, if not tomorrow. Thank you, everyone. Ciao, pessoal. Right.
So we're still zooming around this area. Exploring the
Right,
seep. We're
so-
still on a stay here the whole, and then just move over to waypoint 4.
No, so we're going now north towards waypoint 5.
Waypoint 5.
So we'll
okay.
like, which is right there.
Yep, I see it.
We'll like crossing across this whole patch. So if we see, so like we're crossing and then we saw this sea star,
Okay.
and then we stop. So if you
Okay,
see
so
something
just-
interesting, stop.
Okay.
Stop, but then we're going to waypoint 5.
Okay.
We have worms and the sponge in here,
Right.
and the white sea star.
Okay.
And then
I
here
saw
there's
the
mussels
and the chitin. Yeah, I saw the chitin. Okay. Thank you. Thanks, Juliana. Hi, guys.
Hey.
Hello. So interesting. So we're working our way across the seep up towards waypoint 5.
It's whatever you want.
Yeah.
You're
And then if we
the boss. You're the boss right now.
Yeah, for the time
Whatever
being.
you say, I do.
Oh,
Okay.
okay.
You don't worry.
It's dangerous. That's very dangerous. Yeah.
Yeah, you can.
Bridge over here.
Waypoint 5.
Yeah, go ahead. Good morning.
Good morning. May you please start to move towards the waypoint? Thanks.
Great. Thank you. Good day, everyone. Shirley Pomponi, sponge biologist, Harbor Branch Oceanographic Institute at Florida Atlantic University in Fort Pierce, Florida. And I woke up this morning I turned on the video monitor in my cabin, and there was this massive methane seep area, mussels everywhere. And so we've been exploring. I think this dive started about 8 hours ago, and we still have a few more hours to go. We've been exploring an area that was mapped and predicted to be just exactly what it is that we're seeing. Lots of shrimp here. We just passed over a big sponge garden. We've seen the sponge before on the methane seeps. I'm excited to study it even more. I can take a look at the samples some more. We've seen some amazing worm tubes, but here just massive mussel beds. Heading towards our next waypoint, and we'll stop and take a closer look at anything that we want to explore more. Now we're seeing a lot of shells, remnants of an even more massive mussel bed area. Mapping that was conducted last night by our team of marine technicians and geophysicists, Dr. Rachel Lauer and Dr. Robert Perrin, indicated that there may be mud volcanoes in this area. So we're in search of those areas. Team mapped out different points along the dive that we could explore, and in search of these mud volcanoes. The bottom topography was really interesting from the multi-beam map. It's just contrasting areas of really dark and bright backscatter from the multi-beam mapping, and that's an indication of depressions in the bottom and hard substrate. We started the dive about 8 hours ago and started on a sedimented seafloor, and then we've been climbing up this mountain.
Are we at waypoint 5?
Okay.
Yes.
Sorry.
I'm sorry.
Sorry.
Hi there.
We can also land here
You wanna
to
get
see
down?
the shells.
Okay.
And see what's around.
Okay. Go a little bit.
And-
Just go a little bit closer and just. You want to
Yeah,
land,
no,
or
we
you want to?
want to land.
You want to land? Okay.
Yeah. And we see dead clams. We can also collect some of those. Even if they're
Dead?
dead.
Really? Like just the shells?
Yeah.
Okay.
But if they're clams.
Okay. All right. So we are going to stop here for a little bit. Stop here.
I'm stopped.
Stopping. I'll halt. And then we want to look around and pick up some of the clam shells. Please.
Stop it. Anything of interest?
We want to zoom in the shells and see what's there.
Just a scoop anywhere, or you want something else?
I have nowhere to go, Shirley.
think?
Yeah, we'll
We'll-
see. Maybe just pan the camera.
Yes, go scan around. Okay.
So there's
So that,
sponges.
yeah, the pinkish that you're seeing on the clamshells, actually encrusting sponges, really thinly encrusted ones. We got some the other day, really thin crust, maybe like 2 millimeters thick. So
Yeah, no, that's okay.
That's full zoom there.
Okay.
So all these little patches, the pink, the white, the yellow, those are all really thinly encrusting sponges. Really, the only way you can even get those is by getting the shells on which they're encrusting.
Let's move it again. Let's move the camera again.
Okay.
We just scan what we see.
Okay. Yeah.
Oh, there's another one.
Mm-hmm.
You see this? They're like some tiny sea stars.
Oh, look at that. Hey, can we? Tiny thing there. Is that a little? So use the scoop to pick that up? Would that work? You want to use the scoop to try and see that little tiny sea star? You want to try and use the scoop? Yeah, try and see that.
Or on the biobox? What do you prefer?
What do you think? Quiver or bio box? Probably
If we use the scoop, I
it'll have to be the bio box. If you use it, what do you want to use? You're going to use the
I
scoop?
prefer use the biobox, which
That's
is
fine.
easier.
That's fine.
Okay, keep an eye.
Yeah, look at that. Yeah.
Keep
All right.
an
So
eye on the left.
We will try.
That would be easier to look at.
Find
So the bigger one. Yeah, keep an eye on the big one, right? As I zoom.
you keep an eye on this one. Is that what you're?
Yeah.
Keep an eye on that one. What we want to try and get is that little tiny. What looks like a little tiny sea star. But yeah, if you keep an eye on this, Mike, get that area. Great. Thank you. So, Eagle Eye Bea has spotted this little tiny sea star on this clamshell. And we're going to try and go in with a scoop and scoop up a few of these shells that have all kinds of other things encrusting on them.
Okay, easier thing for you to keep an eye on is the sargassum on to the
Okay,
left.
so that's that
The
one.
sargassum to the left.
And the
That's
sea
it.
stars right there.
And this one here.
Okay.
Because I have to move the side camera now. Right? So
Okay, all right.
that there.
So there's like this,
Yeah.
this.
That's the reference.
Okay.
That's the reference.
Okay.
Right.
Okay. Got it.
Okay, come on.
Yep, I see it. It's right in between. It's right there.
You see anything as well on the sit camera. All right, Mike. All you.
All right, Mike, do your thing. So it's like right
Yeah.
there, right?
Yeah.
Yeah. Ryan, what number will this be? Sample number. 492. Thank you. The ROV has a variety of tools that the pilots can use to sample the animals and the rocks, depending on the size, the shape, the consistency of the target that we're trying to collect. And if you've been watching, you know we've been doing other things like push cores, coring through the sediment, taking water samples, heat flow measurements. So we can equip the ROV with a variety of tools and sensors that enable us to observe, document, collect. The cameras are incredible. They're positioned in several places on Sebastian, so we can see pretty much from every angle around the ROV. Kind of like we have eyes in the back of our head, so we can see everything that's around us. I guess more importantly, pilots are able to use those cameras to position Sebastian, both for moving from one point to the next and also for settling on the bottom and being able to position the ROV to be able to observe and collect some of the targets that we have.
That helps us with depth perception as well. Multiple cameras.
And even though this is a massive area of what were once live clams, they're now supporting a pretty diverse community of other animals that are crawling on them, living on them, encrusting on them. So even though we have a number of waypoints mapped out, our goal for this dive was to search for seepage and fluid flow and bubbles and high density animal communities, including these seep communities that we've been observing for the last several hours, and corals and sponge gardens. And we just passed over, not too long ago, an area where there were quite a few sponges, these white, kind of erect branching sponges that we've seen at some of the other seep areas here. So it was quite a few of those. We got a small sample of that, so we have we're able to confirm the identity of not only the sponges but the other animals that are living here.
Yeah. The red points on the right side.
Yeah.
It's also for
Yeah.
muscles, so we can do that.
Okay. All right.
Yeah.
idea is you want to get
It's part of the
Okay,
solution.
move it to the left, so let's, you know, find it.
got it. Oh,
Same
okay.
thing. Yeah,
Got it. Let me know if you see us at the area that's.
No, it is close. Should be right there. Yeah, it is.
See it?
I don't.
Right
Right.
there.
Okay, Mike. Just, I'm zooming out. Just bring it in the, like, bring your arm into the middle of the. Just point to
Yeah,
where.
it's like right
Yeah, watch where I
there,
shoot it.
right,
Just point in.
right in there, Mike. Right,
There it is.
yeah.
Yeah,
It's
right there.
there, right there, Mike.
See it?
See it? You're getting close.
below.
See it? Right down
Yeah,
there.
watch where she's pointing right there. If you need me to go closer, let me know. Closer? Okay. I need to just reposition the RV there.
You want to reposition? You want to reposition? Okay.
Yeah.
All right.
Alright, bring your arm in.
So we're interested in a tiny little sea star that's on one of these shells. We're trying to position Sebastian so that it's in a better position to be able to use the scoop to collect a few of these shells and the sea star upon which the shell upon which the sea star is living. Okay, there we go. Right there. You got it. Yeah.
Keep pointing at it.
Yeah.
Fine.
Right there.
Just behind the.
Right there, right there. Okay, yeah. Well, I wish I could just use my pointer to scoop it in there. You're getting it,
No.
you're getting it. Yep, yep, closely, right. Just almost.
Hold on. No, don't jump on it. Go to your left and down that. Okay.
Think it's there.
Yeah.
Right, so right in there.
No, no, should come to your left. Right where Shirley's pointing
I
at.
think that's
Yeah.
it. Can't really tell if it's still on there.
Yeah, look, it's right there. It's right, no.
This-
Yeah, right there. Yeah, left.
Oh, there, right there. I see it. Yep.
Right, come
You
forward
got it.
with
You got it. You got
Forward,
it.
forward.
Yeah, it's in there.
Okay,
Okay.
okay.
All right. Yeah. That's good. That's enough. Yeah, it's going to be a bunch of them. All right, we're going to have to. It looks like it may have come off, so we'll just have to be really careful with that bio box and
Yeah, you
maybe.
have a lot of different.
Yeah, that's good. That's good.
So I think we can.
Yeah.
So you put that in the bio box.
Yeah, it'll
keep
be in
the
the bio
scoop on
box.
arm, and we might see another one. I think the place will be full. So don't put the scoop.
Don't put the scoop away, okay.
Exactly.
Yeah.
Have it ready.
Yeah.
You can try with it.
Yeah.
Okay. 14 out. Put it in BB 3B.
3B?
D.
D, yeah.
Delta.
Delta. All right.
Yeah. As well. To your right, hold on. Pressing on the,
Mm-hmm.
come up with the wrist a little bit. No. Come inboard. Inboard some more. Yeah. You start to rotate easy. Clockwise. Yeah. Stop, come left with your shoulder a little bit. Okay.
There's a lot in there.
Okay. Hopefully you find a little star in there.
Okay,
Keep-
yeah,
keep it in your arm.
okay, so, Ryan, it's in
Yeah, slow down, but keep
power
the
box
yeah,
3D Delta. I may have gone to okay, so it's a bunch of shells, but there's a little tiny sea star, which I think really popped off. So we'll just when we search through those bio boxes, we'll have to take a look at that. Okay, great. Okay, you can go ahead and stow the scoop when you're ready, and then I'm gonna move again along this bottom. Across a little bit more and then stop in another area. Okay, so when you're ready, Sonia, just come up a little bit off of the bottom, but stay relatively close so we can search, see if there's anything else interesting on the shells. Okay.
I just need to help me close the porch really started on. The porch is a little sticky right
Okay.
Okay, unfortunate. Yeah, you should be good there. Unfortunate.
It's underneath the porch.
Yeah, sometimes it sticks on it, so it just needs a little nudge on the board.
Yeah, okay.
And normally I would sit on the bottom and push forward.
Yeah.
Not working.
Okay, there we go. There you go. There you go. Okay. Yep. Yep. Mud.
Do you want me still go north or you want me to start moving towards
Start heading towards
number 4?
7.
7.
Yellow 7.
Okay.
Yellow 7.
7. 7? Yeah.
7.
Yeah, sure. Those
Yeah.
yellow ones that you see there are these ones. Like
That's this? Is that this? Is that
Yes.
this point there? Okay. All right. Okay. Some stylaster corals or shrimp. It's interesting how these communities change very close to each other. And why we try and search in a broad area around our target sites.
North of seminary, but there's something
Yeah.
straight ahead of me there.
Yeah, you wanna head towards, because we're north of 7 now. You wanna head back. You wanna go towards.
There's something in
Yeah,
front of
hold on a
me
second.
between 7 and 2.
Between 7 and 2, is that good? Yeah. Okay. Yeah. Yeah. So that just illustrates that, if you continue along like just a straight path, you may miss a completely different community that's to one side or the other of you. So we really try and explore these areas, moving around the target waypoints to try and see if there are different types of communities. And you see now, just a few minutes ago, we were over an area that had lots of dead shells, and now completely different. So if we were just moving literally it's just like with a flashlight moving in like one direction along one line, and if you deviate at all from that, you may see something completely different. So the target points that we've laid out, based on the multi-beam mapping, are areas where suspected mud volcanoes and methane seeps, methane communities. We're seeing now some rock ledges. We just passed over community of stylaster corals, and now we're seeing something completely different. You want to come around to that area? You want to come around there? Yeah. Okay. So you want to kind of come back in that area, right?
Yeah, maybe like coming back to
Yeah.
that yellow before.
Okay.
All right.
Swinging around.
Yeah.
Yeah.
Where are you, Jeremy? We're gonna start to move south, view waypoint.
Yeah, got it.
Thank you.
Black coral, ledges and cracks, and evidence of some worm tubes in these cracks. We're heading towards another area where the possible area of methane seeps, and maybe if we're lucky, we'll see some small mud volcanoes as well. cracks. And the ctenophores. Sponges.
Yeah, let's get to waypoint 4. Yeah, and then in the next quadrant, you can start going south to that 84. That's
Okay.
where we finish one transect like before.
Okay, so
And
when we get to 4, then head towards 84? Is
Yeah.
that what you want? Okay.
and then we start after after 84, we can start going east again.
Okay.
And
Just
there
zigzagging.
there
Yep.
was a big muscle bed there.
Yep, okay. All right.
Any numbers on that confusion or?
There's a little way.
Got it. It's worm tubes.
Okay, heading to 4 now.
Some more stylasters. Lots of worm tubes. All right, so now you want
Head
to
east?
turn east?
Yeah.
Yep, okay, yep. Big crab. Want to take a look at the crab?
Yes.
Yeah, yep. Can we take a look at that crab, get some imagery of that.
The crab, and there's another different eel pot there. You see it? A really
Right,
tiny one.
right there?
Yeah.
Mhm.
So
Yeah,
let's zoom
okay,
on
so we'll
both.
take a look at that.
Look at the crab.
Mm-hmm.
A meal.
Mm-hmm. Yeah, I think there's a little fish over there somewhere. Yep. And then that
Pretty dirty.
fish in there, yeah.
You stop the movement, please.
Like they get close to the fish, they talk to you like. I'll go a little closer to the fish.
Okay.
Yeah, right. It's not just like. So if it is, I think
If you can get closer.
Yeah, he's
to
going
the
to.
eel pot, we can
He's
try
going
to
to try and
swap.
get closer to that.
Yeah.
He's trying to reposition so we can get a closer image of that.
Yeah, image. We can try to.
Yeah, okay. All right. So after we image it, maybe try and suction it.
Yeah.
Okay. That is.
It's a eel boat.
Oh, okay. So just a different one.
Yeah, it's moving.
Yeah.
But we think it's a different one.
Yeah.
From the
From
one
what
we
we've
saw.
gotten before. Yep. Okay. So we're thinking this is
Like there's one
eel
on the
palp.
muscles at the time,
Mhm.
and it's only in between the muscles.
Mhm.
But this one is like.
Yeah. Okay. Yep. Where's it? Yeah, I don't see it now. Do you see it? I don't see it either. Oh, wait, wait, let's look. Wait, maybe.
There it is.
Oh, there it is.
There it is.
You wanna grab your, yeah.
This is an eel pout. We usually see them in and among the mussels. This one's out swimming out along the substrate. We think it might be something different. We're going to try to suction it. It seems to be pretty active though.
Yeah,
It's always a challenge collecting things that move, especially collecting fishes. We're going to try and get in position to use the suction to collect this eel pout. I know this is really challenging to do. My husband was a submersible pilot for the Johnson Sealing submersibles, and that was one of the more challenging tasks that he was asked to do by the scientists. He was pretty good at collecting the fish, though.
No, well, if it's out of move, I'll have to try to.
What we
It's right in the center, the side camera, and the data.
You can see the lasers are 10 centimeters apart.
He's swimming there.
So this is tiny. It's a little fish. All right, we're in pursuit.
It's full.
Suction, 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 suction.
No, don't move
Okay,
in.
don't move. Okay. So Sonny's trying to get us in
I'll turn my
position.
head in.
Okay. Yep.
Still coming
Got
down.
him in sight. Right
Yeah.
down there.
Okay, bring your arm down a little bit.
So close, so close, so close, so close. Ah, so close. He's like, what is that? He's.
Seen him in
Do you
there?
see him, Sonny? I don't see him. Oh, there he is, down there. Right down there.
Yeah. Got it or doesn't know?
I don't know. That's something. That looks like it might be something else. Is that? That's something.
Yeah, it's
That's
something
something
else.
else, right?
Now we are seeing eel boats everywhere.
Alright, you don't want that one.
Eh, you want that or not? No.
Might be a
No.
bit different.
No. Let's look. No, that's something else.
Alright, keep that in your mind if we see it again. Alright, which way do we need to go now?
Yeah. Okay. Went ahead. East. There. East. Yeah, that's a different one. It's like
Yeah, these are red tails.
there's rat tails. Yeah, rat tails.
How far east do you go?
Sorry?
How far east do you go? Just for
If we follow the same track that we did before, we will find the other mussel bed.
Okay, you guys want to stop more or less somewhere here around the waypoint center.
Yeah,
of the-
probably up there. You see that?
Okay, here more.
That part? Probably there.
Can't do it, so. Thank you.
Maybe we can stop in this
And that
graveyard to do the same thing that we
right
did
down.
before.
You want to stop right in here, or
Yeah,
up?
or
Up
these ones.
over here. Okay, so Sonny, we want to go over here, settle down.
Yeah, see if we can find another of those sisters. Now that we have the scoop
Yeah.
ready.
Who's that? Is that a clump of worm tubes there? I can't tell. Yeah. Okay, then 1504. Some anemones. Bryozoan maybe. Encrusting sponges. Getting some close-up imagery of things that are attached to these shells.
sponges
Yeah.
here, right?
Hmm?
Fewer sponges, less sponges.
Mm-hmm. Mm-hmm. It's just every now and then occasional sponges. There's that pink one up there. Okay, yeah. Can you take it?
Yes,
Yeah,
of course.
that defense, yeah, yeah. Okay. All right, so we're going to continue moving along this area of these dead clamshells and continue towards our next waypoint. I'm going to end a few minutes early. I actually have a thesis defense I have to participate in a couple minutes, and turning it over to Bia, and she'll be with you for the next hour. Okay, see you tomorrow.
It's actually nice.
Yeah, the stop. I'm ready. Okay, we have only eel pouches already.
Oh yeah, but I think that's the one that is always on the mussel beds. So this is, I think it's not the guy we are looking. The other one is like grayish.
Okay. No, not that one.
Yeah. Thank you. I think the other one is gray. Hello everyone, Beatriz Naranjo from Costa Rica, collaborating with the University of Costa Rica and supporting species and all the science team here on board Falcor 2 while we keep exploring Trinidad and Tobago. We found a large methane seep today, probably the biggest mussel bed that we've seen during the expedition.
Tell me when
Let's
to
take a look.
stop reading, sir.
Okay.
What do you want to look at?
The coral, these like dead worms, and then we can start going south.
Zoom in there now. To the rims.
Yes, it's the whole rock and the coral. The coral, and then we can go south. Okay.
What's that part of ours?
Hello. This, you say? Hi, Zach. How are you?
Amazing. Yep.
So we're hitting the place that we found earlier. Remember that we were finishing a transect, then we kept going, so we are heading to that site to do the same thing.
Oh, the other mussels.
Yeah.
The other mussels.
Yeah,
Okay.
so we will do the same thing.
Okay.
Like flying around the edge to have an idea of how big it is.
Yep, copy that.
Thank you.
You know what, Sonny? I got something if you're bored for your designer brain.
Oh.
So, that high temp probe, but the idea is cool with the magnet, but the execution just kind of sucks. You know? I wonder if we could do it in a way where it's never in camera view, and it's still not hard to get. It doesn't have to be magnet. We used to have a funnel, whatever, but there's something that's, don't you think that thing kind of sucks? It's always falling out.
Yeah.
I was even wondering if we could put it on the side of those, past the quivers or something. I kind of like we do the BBC light, but that way it's always out there. If you had time. All right, all good?
Okay.
Station, keep on alt is on it.
I have this and on the right arm I have this pen.
He's so ready.
You're ready to go.
He's really ready.
Copy that. Ready to party. Something like it.
Okay, so we can go south and try to hit the other place that we found earlier.
Start the direction of waypoint 5.
Yep.
Yes.
Okay. No.
Where are you, Gerald? We're going to start to move south slowly. Change your waypoint towards the south.
Over there. Thank you.
Thank you.
Slowly. Speak for yourself. Wow, some cool stuff here.
Yeah, it's been really interesting.
Good size of this rock.
Buenos días a todos y todas. Mi nombre es Beatriz Naranjo de la Universidad de Costa Rica y estamos explorando un emisario de metano a más de 1000 metros de profundidad en las aguas de Trinidad y Tobago. Este ha sido el sitio con más mejillones y cubriendo mayor área que hemos visto en toda la expedición, y ahorita estamos intentando acercarnos a otro de los sitios principales de mejillones que observamos temprano. Wow. Can we zoom in on this, guys? Nice. Yes, please. We're just at the top of the world. Beautiful.
Little garden, huh?
Yeah.
Sea spider right there.
Oh, yeah.
It looks like if I just bumped it, it would fall. I'm sure it's not.
No, probably not. Cool. A new screen background
We just take the
for
whole
my
rock.
computer. Yeah, that's it.
2 ounces.
Yes,
Carry on.
beautiful. You have them? Yeah. Yeah, we actually collected this coral maybe yesterday, the yellow one. Yeah, yesterday. It's a spider on the center. Really nice. Let's take a look.
Bottom left.
Yes, this here.
That guy.
Yeah, but I don't know if the same one we already have. We can bring the what? But really early in the expedition, so you can get the snip. Yeah.
You want this?
Yes, please. Snip,
All right,
snip.
let me think about where I can park for you.
No, with the. So yeah, you have to put away the, I don't know, the scoop. I think that's what you have on the left side.
This one's like kind of a weird pot, can you let go of the suction so I don't pull it out your hand there?
We'll slow it up.
Yeah, you can stow it or just drop it. Yep. You can stow it or drop it. Either one.
Estamos observando estos corales en la cima de esta roca, un pequeño jardín, y la mayoría de estos corales ya los hemos recolectado para tener muestras que nos permitan observarlos. Creemos que el amarillo es algo cercano a un acantogórgide, para quienes nos están preguntando, y el blanco es un primnoide. Tenemos también un coral negro y algunos hidrocorales. Así que una muestra de la biodiversidad en una pequeña roca.
Ramming out. And we're going to have to put down the scoop as well.
Estamos guardando todas las herramientas con las que el robot estaba preparado.
These prim nodes, are these the ones that break super duper easy?
Yeah, they are not hard to collect.
They are hard to
No, I mean
They're the ones you
they
touch and they
break.
just. I'm just wondering,
No,
since he has
not like
this-
that.
Not like that?
Not like that.
So he could grab it.
Not exactly. Like
Okay.
something in the middle of that and the black corals that are possible to break.
Fred, Jarvis, you can probably move.
Yeah, just roll a couple of
Thank you.
So we'll try to land here,
Alright, you gotta put the scoop away though, right? Pull it
trying
down.
to put the scoop again on the RV. We were super ready with a lot of tools outside, so we are trying to save them and collect a sample of the primnoid coral. It's a kind of octocoral, the white one that we are seeing just in front of us at the top of the rock. Were you here for the toadfish?
No, I heard about it though. I
Yeah,
love those.
same. No, I was not here, but I saw the
Those are always
pictures.
cool. Those are always super cool. Where's this gonna go? Quiver, Bob?
In a quiver, it's okay.
We don't have anything in any quivers, right?
No, not yet.
Alright, so you go ahead and open the quiver. Any quiver, dealer's choice.
No. One is okay.
Is that the same thing right there? Because that one might be a bit easier.
It looks the same for me. You agree?
It's okay?
Yeah, this is better. So we'll try this one. We believe it's the same one that we had at the top of the rock. Y para quienes nos preguntan dónde está el robot Sebastian en este momento. Estamos a 1028 metros de profundidad explorando una zona al este de la isla de Trinidad.
Okay, I'm fairly lightly perched. It's kind of a terrible view for you, other than
Hemos estado en expedición todo este mes y probablemente hoy sea el último buceo que hacemos por debajo de los 1000 metros.
So I'll just be very cautious.
How big do you want the size?
I think it's a little better view.
Something like this would be good. 10 centimeters. Something like that. Like, maybe you can grab this at the end, at the bottom. Yeah.
You can get the top.
Oh, okay, the top.
Yeah, you just top's
Okay.
the only place you can be able to get.
All right. So yeah, something like
But I would
here.
just try not to touch it until you're ready to grab, because if it is one of the crumbly ones, it'll go. That weird thing to the side of it that comes out of the rock, goes up and goes back into it.
Yeah, I think there's like 2 different colonies actually.
Is that like a handle?
Mm-hmm. Ah, this thing is.
It's like a door handle, huh?
Yeah. Should we open it? One.
It could possibly go off. Probably the plug.
Yeah, a bit down. Sorry.
Not too much down.
Not too much. No, not down. Sorry, closer to the rock. I mean. Yes. Thank you.
Hey, you want a glam shot?
Yes, please. Nice background also. What number is this, Ryan? 493. Nice. Thank you. Yes, please. Thank you. Algo interesante que hemos estado observando estos días, explorando los emisarios de metano, han sido los tiburones. Hemos visto, diría, al menos 3 especies, incluyendo tiburones gato y tiburones gatos falsos. De hecho, hoy, más temprano en el buceo, vimos una cápsula, un huevo de uno de esos tiburones en coral negro. Y también hemos visto muchas quimeras, diversos individuos cada buceo que hacemos.
Okay, there you go.
Yes, please. Thank you.
Does that BBC handle light in the starboard box? Is that still tear up on the side, you know? You know what I'm talking about?
It says BBC light. I think BBC will look at
So this dive was planned for 11 hours, but we are actually extending it, probably, because we found these big mussel beds, and we need some time to document them correctly. So we are planning to stay a bit more, probably.
Am I heading for waypoint 5 or am I heading for that big
When we collect these samples, we do it with the purpose of being sure what we have there, so they are like witnesses of the biodiversity that we are seeing. In most of the cases, it's really hard to identify the animals just with the video. In some cases, it's possible, but for places that have not been well explored or documented, it's really hard to put a name on all the animals that we are seeing. So that's the reason why we sample. Yes.
Am I heading for waypoint 5 or am I heading for that big second mussel?
No,
field?
the second mussel that we found with you.
Okay.
Which I believe might stand.
See that little loop? Should be right
Yeah,
there.
exactly.
See that loop on my screen? You don't have it from the trail? Yeah, that's where I'm going. Oh, boat's fine. Boat's fine over there. Boat is fine.
Yeah, here it is.
Alright. What you want to do here? Transects, let me guess.
Yeah, but probably first it will be easier if we just fly around the edge of the living mussels and do the same thing that we did there, actually the waypoints.
problem. You know how to make a new mission, Mike? Here. Create mission. Second. A weird color that nothing else is. Okay. Alright, and then when we go, we'll just do the waypoints after. Alright.
Okay.
I'm ready if you guys are ready.
Okay. So Ryan, we will start flying around. See the extent.
Just at the end we'll do it. Don't worry about it till the end. We'll just use the snail trail until then, okay? Alright, I'm just gonna try and stay right where the dead muscles start, right?
Y ahorita lo que vamos a hacer es volar sobre esta línea que separa las conchas muertas de las conchas vivas para tener una idea de la extensión de este campo de mejillones. Para eso estamos haciendo una misión en la pantalla que tienen los pilotos, y con este vuelo sobre esta línea vamos a poder estimar el área . Una vez tengamos esta área, podemos hacer transectos que empiecen lejos del borde que separa las vivas de las muertas y poder hacer estudios de zonación. Hemos visto que conforme nos alejamos de estos sitios empezamos a ver corales, conforme nos vamos acercando empezamos a ver gusanos, esponjas, hasta llegar al sitio principal de los mejillones. So what we're doing here is try to measure the area, the extent of the mussel bed. So we are just following the edge, and at the end we will have an estimate of the size of it. And afterwards we will conduct some transects to have an idea of the transition between the mussel bed and the background area. We've been seeing zonation, so we have corals, and we start to see tuberons as we approach to the heart of the mussel bed. So we will be flying afterwards in straight lines, using our lasers to have this more quantitative information. They almost touch each other.
Oh, yeah.
20 meters.
There's one in between, you know?
Mm-hmm. Probably.
I wonder how many there are like see the whole sea.
Oh.
Is it just covered in these things, or
Mm,
is this it? Oh, what is this?
it's a pile, probably.
I wonder what animal this is going to turn out to be.
What?
The shape of the mussel bed.
Mm. What do you think was the first one?
Water hose Definitely.
I told Juliano it was a upside down boot.
Yeah, I could see that. See that. It's kind of got a broken wheel right in it.
True.
Too much walking.
So far, it looks like a seahorse, the head of a.
Maybe it'll be a pair of boots.
That would be fun. Yeah. It will be easy to name the place if that happens.
What would you name it?
Pair of boots. Las conchas blancas que vemos son las que están muertas y las que están vivas son las de color café. So we're following the edge between the living and dead mussels, or just the. In this place, we don't have a. It's not so different, but the white ones are the ones that are dead, and the brown ones are the ones that are living.
Two worms mixed.
Mm-hmm.
Seems like that's a little bit rare, huh?
Yeah. Y la razón por la que son blancas es porque están hechas de carbonato de calcio, que tiene color blanco.
With that, Mike? Boo. All right, I'm going to take my turn now.
Okay. Look, a seahorse face, like the snout.
Oh, yeah, okay. I can see it.
Is that the end of the flaring?
Yes, that was the end.
So, this is another one?
This is another one, but it's really close to the one we were.
And
So actually,
have you seen have you been
yeah,
this one?
yeah. It was not. I mean, there was no patch up there.
But were there any mussels in that?
Yeah.
Oh, there were.
Yes, like more
Patches.
patches. Yeah.
Cool.
Yeah, it's cool. You see the seahorse face. I'm glad you do.
Yeah.
Zach says he does, but I'm not
He's
sure.
lying.
Probably.
Yep.
He hasn't made a joke. so
Whatever you
I'm
say,
not
lady.
He's just a driver.
So see Uber.
So I would do the same.
Yeah,
like
sounds
that
good.
transect.
You get anywhere near those lines, you just don't want to do it.
Y parte de la razón por la que estas mejillones mueren es porque la fuente de gases emergiendo y por de la que se alimentan gracias a las bacterias que tienen dentro y con las que mantienen una relación simbiótica. Esas fuentes de gases se cambian de lugar, se apagan. ¿Dónde?
If we have shimmering water, can we just try to take a temperature measurement?
If we see, sorry?
Shimmering water.
Yeah, I haven't seen.
Oh,
Have you?
apparently they just said they saw.
Oh, really?
There was some this morning somewhere, yeah.
Maybe by the muscle patch.
Okay, we'll keep my app.
We'll just check by the muscle patch, and if there is, we'll just try to get a temp
Okay.
measure there.
You guys know, do we have an updated off bottom?
I'll go check with them. We're still going. We were originally meant to come off bottom at 10, but we are not sure yet.
Too much cool
No.
stuff.
No.
Definitely lab on it.
Okay,
No?
am I hallucinating?
You saw it?
I don't know.
You saw.
There's some earlier this dive, so.
Yeah, I saw it earlier and thought I just needed to go back to sleep.
They probed it earlier though, I think, but if we see more, we should definitely probe it.
Yeah.
What would that shimmering be?
Warm water.
Okay.
It has to be. The shimmering is from different density water, and the only way you get that is warm water. Yeah.
So you saw
So,
it just
Yeah, I
now?
saw it. I mean, I thought I was tripping, and then Rob saw it too. But I saw it
Okay.
earlier today.
Doesn't mean
I-
you're tripping.
I mean, I'm sleeping.
Okay.
But we're still not sure if that's going to be when we come off bottom.
Okay.
Maybe not. Can we zoom in on?
Sometimes the camera, like,
I know, and that's why I'm like, when I saw it earlier, but then,
I mean,
but
but it
Rob
can
saw
be-
it too.
I mean,
Like, it's like, no, two people saw. I know, sleep deprived people thinking things are shimmering. It's very funny.
and hydrogeologist.
Hydrogeologist.
Just wanted to
I wanted to
it.
see shimmering water. I
Yeah.
manifested
Okay,
in my
but I will
brain.
keep my eyes open. If you do, like,
Okay.
scream.
Okay.
Scream.
Scream? Okay.
We will do some transects now, so
Okay.
we
Look
might
how
see
many
it.
waypoints. Look at that, guys.
Yeah,
That way, I
beautiful,
think we
right?
can export them for y'all then.
Yeah, that
Or
would
at
be
least they don't
amazing.
disappear like the snail
Yeah,
traps.
actually, let me take a
So we got
picture.
that and that.
That is awesome.
Can you zoom
Wait,
in
when
a
did
bit?
you start? Look at this.
Zach.
It's so cool. Zach. I love it.
If you zoom in, you get 10-meter grid.
Yeah.
Bleep, bleep,
Is that
bleep,
10? No, that's 1 meter, right?
10.
There.
No, 1, sorry, right. So that's 10.
That's that.
That was the worst.
It looks like Pac-Man with this mouthful of
Just use Alex, he's like our AI.
Okay, so now we will do the same
Maybe
as we
it's
did
easier.
earlier.
I have to ask him a question.
Like probably at
It's big.
transect here. It would be interesting if we can get to that 84, just because that's where we end the other transect.
Okay. So last time it seemed like you didn't really care about 100 meters anymore. You just want to go from the
No,
start
yeah,
to the end.
in this case, I think it's not.
Is that fine?
Yeah, it's
Just start to
fine.
end. Okay.
Yeah,
So I'll just flash the lights at the beginning and end.
yeah. So
I'm not
we-
really sure of the topography, so I don't know
Oh,
if it's
yeah,
going
that's
up or
true.
down, but we can try from up here since we're already up here if you want. And then if it sucks, we can do that.
Okay.
You can do that.
Yeah, so we
Like
can
this,
get
the biggest area.
yes, exactly. Yeah, seahorse. It's not. Was more like a seahorse for the waypoints.
Really?
Yes.
Oh.
I mean, just the head.
What about
of
a
the
cat?
I see it. You know what it is? It's a sock to go with the boots.
Oh, I see it.
It's a dirty
Yeah,
sock.
there's That's it. Good job. You're on our wavelength. At 1 a.m.
Yeah.
we see cats.
So yeah, I do think we have to go
We need
north.
Timothy. I'm with you.
Yeah.
Timothy. I haven't seen him yet.
I know, we need Timothy seat.
We do.
Then you're done.
Can we name the seep, Jim?
You don't even include yourself.
Yeah.
All right,
Zach, I think we have
yeah,
to go
farther back.
north. Yeah.
Up.
It might not end, but it will be interesting to start closer to 84 because that's where we
sorry.
finished the other transect.
84.
Bridge, new waypoint.
Your team, biology. I like
The
that.
biologist.
Biologist.
Thank you for
Yes.
this.
You got a new branding.
You're so proud of Juliana. It was
I
you?
know, she was.
She was?
She just did that.
You must
Sometimes
be.
the best things happen that way.
Yeah, you must be so proud.
Dad jokes.
Is that the
Dad
first
jokes.
time you've gotten that?
I love it.
Oh, we're trademarking that.
Yeah.
100%.
We're getting stickers.
Yeah.
Yeah.
Really, you never heard that?
Oh, I know, and also like, yeah, patchy sleep.
Oh. You're baking bread and it smells. That's what I smell every.
Hey, how did the death sponges come up?
Oh, guess what just.
Oh, really, really good. Yeah. Yeah, we got 2 carnivorous sponges last afternoon, yesterday. And yeah, it was really interesting. We also got some worms attached to the sponges. Yeah, some amphipods. No, sorry, the worms. We lost the amphipods that were attached to it, probably in the collection process.
Did you figure out what the little things were inside of the sponge that were thinking?
No, but I mean, we are trying to figure it out. And Shirley thinks we saw different stages of like when they grab something, probably it inflates and englobes what is eating. So it was interesting. A lot of questions, more than answers, which is always good.
Did we do the cross transit?
We are trying to. So we are in that point that was close where we will finish the other one. But still like a lot of.
Stuff going on.
So we did one on the wood, right? Top, like this one.
Obviously.
I see upside down. And we put up that white where we are right now, but we actually have to go even easter because
Where
yeah, we have this is 84, and we have to go even farther away.
Yeah.
So I think we should do the same right now. Just go north, start seeing, because there's a lot of worms. So it will not be a transition. So my question is, do you think we should go north until we have worms?
Sorry. So on this map, there is sorry guys, give us 1 second.
We did one transit here. And I did that here. I want to do now this one.
Yeah.
So we are here, but we still have worms. So yeah, but the thing is like we want to see
can go further. So we're just gonna lower it. It might never dance. We'll just drop off the edge.
Okay, so I think we should just keep going north until we don't have worms. If that happens, if we see that it's too long, I think there were no worms like closer to 8.7, so we can try that to actually have the whole idea of the transition. Yes.
Over to you, Gerald.
You wait for a couple.
Thanks a lot.
All right, just say uncle, okay? What's that? We're going to.
We're going to,
But
yes.
not yet. It's not gonna
Okay,
be
you,
just
yeah,
start and end.
yeah. Let's do it here.
Okay.
We still have worms, but they will be everywhere.
Right, Jarvis, you can probably move.
Balls in the mold.
We're gonna do a transect to the south, approximately 100 meters, maybe a little more, so we'll give you another move in a sec. Okie dokie, everybody ready?
Yes.
Okay.
Yeah.
Once I get about 20 meters away from them, Mike, you can give a nice slow move to the south if you want. 0.2.
Say it again, sorry.
I was talking Mike.
Ah, okay. So we're starting a video transect here. And our idea is to, so we were like away from the main mussel bed to start seeing the transition. As we approach, we will cross it and then we will keep going to have all that changes documented.
Hey Mike, which one's missing the diffuser right now? The upper starboard?
It's missing, if I'm not wrong, it's missing the upper port side.
Upper port.
Not 100%.
Wow, do you see that green line?
Yeah.
Yeah.
Oh, there's one of the eel pouts. So they're making fun of us.
Yeah. It's like,
I thought we got one earlier this week.
He or she baited.
Oh, you want to catch one?
We tried earlier.
I bet we could get one.
No, not on this dive, but when we did the
no, but
suction earlier.
Yeah,
Oh,
we could,
yeah. No, this is a different species.
All right,
Yeah.
perfect.
but you'd probably have to suction that.
They're
Huh?
very cute.
we did a suction today besides the attempt to catch this, no, right?
More of that green there.
Yeah.
Yeah.
The spots. Mold. It's moldy.
Sorry, Ryan.
That's 20 meters from the red zone.
Sure.
So,
Okay.
there.
Morning, Geraldine.
So you start to see a lot of
Yeah, go ahead.
tube worms,
New waypoints,
serpulids,
please.
all these hydrocorals,
Thank you.
and tube worms start to appear. Lots of sponges as well. Got all this.
Oh, weird, look at that.
Yeah. It's long stuff.
Yeah, the seagrass
Yeah.
looking sponge thing. Did we see that at the first place?
I don't remember this. You see this, Ivan?
You remember I thought we saw little
Can
fields
we
of
drop
that.
it just a waypoint and then we can always come back after?
Yep, you want to drop a marker there, Mike?
What's our marker? Is it bacterial mat?
I was curious. Look at this weird
Seems
long
like that. I
bacteria.
don't know. Mm.
There's some more right here.
Mm-hmm.
What's the name of this marker? Very good.
Hairy.
Are you sure? Okay.
And mussels.
Zach,
Yes.
when we were chatting down our beach yesterday, you weren't wearing a hat, is that right?
Correct.
Can I,
I don't wear a hat any of the time when you do it.
can I get Mike to wear your hat?
I
Or is
think
Mike got his
he's
hat?
he's got one.
Where's your hat?
Huh?
You got your hat? Where is it?
I can't know that.
So I'm just taking advantage of today you sitting on the lows or grab some
I
shots.
can go there, please.
Yeah, go for it, Mike. We're good.
Cheers. Cheers, Mike.
Just gonna rename that.
Starting to hit the boot here. Or the sock, sorry, the sock.
The sock, yeah.
Right in the toe. In the stinky part.
Wow.
Oh wait, but we were here before.
Yeah,
this
we
is
were.
We're
Yeah, this is
crossing
the
it
start of
We
it.
went away to
Yeah.
the north.
north to south.
And we will cross it to the south, and then the same, but west, east.
I like it. Everyone says that, right? Exactly.
Ready?
Mm. crabby.
Just about the middle of the sock. This is the thin direction too. If you went the thick direction, it'd be even longer. What's going on here? That's all new muscles, huh?
Yeah, like baby muscles. Right where the hydrates are. Yeah, it's interesting. It's like really patchy. More tube worms.
Super interesting.
Yeah, it's all covered.
I'll leave my hat on or take it off?
That's easier to get first.
Just tell me when you're happy.
Yeah, we have to keep going.
Oh yeah, I'm keeping going for sure. Stop it, you're gonna stop. Here's fish down there.
Oh yeah, that's the one we need.
Are you looking for that?
Yeah, it's a different one. It's an eelpout, but it's we believe it's different from the ones that are living with the mussels. That one is white. This one is gray, and they are like the surrounding area,
Oh,
not
they
on the
don't live in
mussels.
Not in the mussels.
Exactly.
Yeah, I don't know how far we're gonna go, so might as well just keep it rolling. Oh, look at that line.
Yeah.
Just a heads up for folks, we're going to be restarting the stream here shortly. So keep an eye out for part 2 if you're over on YouTube and you don't get kicked over automatically. See you there.
Mm-hmm.
Whole thing where
Yeah.
all those tube worms are.
And all those tube worms are taking advantage
Oh,
of
there's
that
mussels in there,
fracture. More mussels. So yeah, the gas is escaping here
It's like stitching
in
it all
this fracture.
Maybe kind of starting to end here.
Yeah, but we still have some tube worms. So let's just do a bit more. until we find another one.
So far, just a bunch of shells.
Yeah, we find another one.
Yep.
Look at that.
No, you did it. I think that's a small one.
Mine never ends.